Hello, everybody. Welcome to the stream. Hello. Let me go ahead and say hello to everybody. Hello, Dreamless. Hello, Astral. Welcome. Hello, Bobo. Got you set up in the lab. Hello, everybody. Hello, Bobo's lab. Tube labeling. So tube labeling isn't that bad if you've got those sweet fine-tipped lab markers, which I always lose, and therefore default to the regular fat tip markers, which makes labeling a nightmare. Hello, Seed. Hello, Writer. Hello. Welcome, Ajmal and James. Welcome. Goldscope. Welcome. I don't know if I said hi to you, Seed, but hello, Seed. Today, what we are going to do is we are going to be doing something that I was always planning to do, which is to read a paper about measles. So for those of you who don't know, I'm moving towards having these streams be more convenient, I guess you could say, for my content pipeline, because I'm already going to have to read this paper for the measles video, which I do believe is probably going to be the next video I do. Hey, Mylan, welcome. So we're going to kill two birds with one stone. We're going to make some content by reading this paper, and we are also going to be preparing eventually for a measles video. So I'm kind of interpolating the measles video pipeline with the current video that I'm editing, which is the long-term E. coli evolution video. And so this is a way for me to break up the kind of flow of my day so that I'm not just video editing 24/7 on the same thing. Change it up every once in a while and read something else. I find that that's probably a lot more sustainable with regards to keeping my sanity as I navigate the world of content creation. All right. So without further ado, let's get right into it. This is a paper from PLOS One, my beloved PLOS. This is a fantastic journal that has all of their articles open access with very permissive copyright rules as long as the sources are cited. And so we are going to be taking a look at this particular paper, cytosolic 5' triphosphate ended viral leader transcript of measles virus as activator of the RIG-1 mediated interferon response. And the reason why I chose this paper is because whereas the flu virus has, encyclopedias worth of information, the measles virus actually isn't that well understood, which is really surprising because, we kind of mostly got rid of it with vaccines. So I thought that we were going to come into a virus that is very well characterized, that we know a lot about, because we were so good at getting rid of it. It turns out we don't know a lot about how measles functions. And I think that's because vaccines are just that damn good. Why bother putting money into measles research when the vaccine is so incredibly effective? Like the approach of a vaccine is actually uniquely good for measles, which is something that I'll get into the video. But for now, I'm still doing my research around this virus. So it was really surprising to me that when you Google Scholar the measles virus, it is actually pretty difficult to find virology information about the measles virus. You mostly get articles that are trying to engineer the measles virus to kill cancer. That's like the hot thing to do with the measles virus, apparently, which I think is kind of interesting. But yeah, it actually is really bizarrely difficult just to get the basic information that I need for a Phi video, which is unusual. I think most of the targets that I choose to talk about are kind of famous or well covered in that kind of regard. So today we're actually going to be taking a look at something that's relatively basic about this virus, and it's how does the cell detect the measles virus. I did a little bit of pre-reading because I wanted to make sure that the article I chose was interesting. So I think that this is going to serve as a pretty unique entry point into understanding measles virology. And this also gives you a kind of idea of what my research process is like at the very beginning, right? Because I've read, I think, 2 review papers on the measles virus, but no like hardcore real paper quite yet. That's pretty cool. I'd never considered that something being so well contained would be to lower depth of knowledge. Yeah, that's just like the money isn't really there, right? Isn't it Rig-I, not Rig-1? Oh, is it? I'm not sure. I've never actually heard it spoken out loud. Hello, Artistic. It's Roman numerals. I actually don't know. I think it's rig 1 for interferon type 1, but we'll see. I'm coming at this from an immunology amateur. All right, let's get into it. So let's read the background. Double-stranded RNA, dsRNA, is widely accepted as an RNA motif recognized as a danger signal by the cellular sentries. Love that wording. However, the biology of non-segmented negative strand viruses, or mononegavirales, is hardly compatible with the production of such dsRNA. Okay, so to explain this, right? So when we talked about the flu virus, we talked about a segmented genome. So if you watched that flu video, you're very well familiar with this idea. The flu virus packages its DNA as different pieces. And so when those pieces are or when those pieces are converted to a form that is usable for the cell, you often get an intermediate that is double-stranded. And so that double-stranded RNA almost never happens in the natural context of a eukaryotic cell. Double-stranded RNA is often the result of viral infection. However, if you have a virus that is negative-stranded, so this is just a way to explain that this RNA is ready to be made into protein immediately. It doesn't have to be converted into the usable form of RNA first. It's ready to go. And so it never really actually forms a double-stranded RNA segment. And so this in of itself is capable of subverting some mechanisms of viral surveillance. So I think that is pretty interesting. Why does something want double-stranded RNA? Why not just use DNA? It just kind of depends on how certain viruses evolved, right? There are all sorts of benefits for being a DNA-based virus, and there are all sorts of benefits for being an RNA-based virus, namely that the RNA viruses appear to mutate a bit faster than the DNA viruses. Okay, methodology and principal findings. During measles virus infection, the interferon beta gene transcription was found to be paralleled by the virus transcription, but not by virus replication. Since the expression of every individual viral mRNA failed to activate the interferon beta gene, we postulated the involvement of the leader RNA, which is a small, not capped, and not polyadenylated RNA firstly transcribed by this class of viruses. Okay, so let me translate that a little bit. So interferon beta, so this is an immune response. It's an immune program that is activated upon the presence of a virus. So it looks like that when the virus is transcribed, so when its genetic material is copied over, but not by virus replication. Okay, I'm a little unsure as to what this difference is now that I read this out loud. So we're going to have to come back to that idea. I'm not ready to talk about that yet. But the next sentence is, since the expression of every individual viral mRNA failed to activate the interferon beta gene. So basically, I'm guessing this means that they tried to inject cells with every individual possible measles mRNA to try to see if, what target is the cell trying to detect when it comes to a measles infection. Apparently, if you just give the cells the viral mRNA, it doesn't appear to be activating, doesn't appear to cause an immune response, which is kind of interesting. All right, we postulated the involvement of the leader RNA, which is a small, not capped, and not polyadenylated RNA, plays a role. So, okay, so I've vaguely heard of leader RNA, but I'm actually not super sure what this is. So I'm hoping that we'll learn about this later on, and if not, then I'll just Google to keep myself up to date, right? Sometimes you don't really get the privilege of having the you don't really get the privilege of having them define the stuff for you, but we'll just see. Which is a small, not capped, blah, blah, blah. The measles virus leader RNA synthesized both in vitro, so in a dish or in a glass tube, and in vivo, so inside a cell or in an organism, was efficient in inducing the interferon beta expression, provided that it was delivered into the cytosol as a 5' triphosphate-ended RNA. Okay, so this, whatever this leader RNA is, has to be modified chemically in a very certain way, and then this is the molecule that the cell uses to detect a measles infection and to be able to respond. The use of a human cell line expressing a debilitated RIG1 molecule together with the overexpression studies of wild-type RIG1 showed that the IFN beta induction by virus infection or by leader RNA required RIG1 to be functional. Okay, so the question that this paper seeks to answer is, what is the mechanism by which cells detect a measles infection? And so how does that happen? So we know that the cells are likely going to detect whatever this leader RNA is, and then it is going to respond using this pathway called RIG, and we'll get more into that later on. I am passingly familiar with the RIG pathway, right? I know it's an immune pathway. It's interferon, related to interferon, which prevents viruses from replicating and spreading and all that good stuff. So we'll learn as we need together as we continue. Okay. RIG1 binds to leader RNA independently from being 5' triphosphate ended, so it doesn't matter if that leader RNA contains this chemical group, while a point mutation Q22299A predicted to establish context with RNA fails to bind the leader RNA. So this paper is going to identify the exact place on that RIG1 protein that is important for measles RNA detection. Since the 5' triphosphate is required for optimal RIG1 activation, but not for leader RNA binding, our data support that RIG1 is activated upon recognition of that RNA end. Okay, significance. RIG1 is proposed to recognize this family's transcription, which occurs in the cytosol, aka the cell body, you know, the guts of the cell, if you will, while scanning cytosolic RNAs, and to trigger an interferon response when encountering a free 5' triphosphate RNA resulting from a mislocated transcription activity, which is therefore considered as the hallmark of a foreign invader. Okay, so pretty heavy abstract. I'm going to try to put it in my own words as often as possible, but we will see. We will see. What is transcription in this context? I'm trying to get a sense for that myself, right? So transcription is by definition just the conversion or the reading of DNA to make RNA. But this paper is making a distinction between viral transcription and viral replication. I'm guessing replication includes protein production as well, which is why this is separate. So let's see if the introduction can shine a light on some of these headier ideas here. Okay. Okay, let's start at the introduction here. The cellular innate defense is initiated with the recognition of peculiar data. This paper is written kind of interestingly. I wonder if it's because this is like a English as a second language kind of situation. The cellular innate defense is initiated with the recognition of peculiar danger molecular motifs called pathogen-associated molecular patterns, PAMPs, by the pattern recognition receptors, PRRs, which result in the induction of a type 1 interferon response. Okay, so, you know, we're talking about innate immunity. This is the older variant of immunity. This is the immunity that most, no, I shouldn't say most, this is the type of immunity that does not require learning, right? This is simply stuff that is generally recognized as foreign versus stuff that is generally recognized as self. So there's no T cell response here. There's no B cell response here. This is just the body has evolved to keep track of stuff that generally looks dangerous. So that's what this pathogen-associated molecular pattern is referring to. How do you pick which papers to read? Well, first, it has to be open source and copyright enabled, so I can legally have it on my stream. And two, I wanted to pick something that was interesting about measles, but not the most interesting thing about measles, because I don't want to spoil you all for the actual video. So I read the abstract and found that this was, I think, pretty unique for a virus to take advantage of. So I wanted to find something that was measles specific, but not like the best stuff, so that you actually do watch that video. All right. Okay. Unless you're talking about the actual process, in which I just use a combination of Cell, Nature, and PLOS, and Google Scholar, and I filter by copyright permissions. Okay. In the cytosol, the helicases, RIG1 and MDA5, induce interferon transcription upon recognition of viral double-stranded RNA. Oh, I didn't know that RIG1 was a helicase. So RIG1 I just know as a protein that activates the immune response. I didn't know that it existed in the class of enzymes called helicases. So helicases are enzymes that unwind DNA and prepare DNA for transcription, replication, etc., etc. Okay, so this is a molecule that will enable the production of interferon at the DNA level. Cool. I understand. Okay. Mononegavirales, it's probably about mononegavirales, are characterized by having their RNA genome and antigenome tightly encapsulated by the viral nucleoprotein N, making them resilient to silencing by siRNA. Okay, so characterized by having their RNA genome and antigenome. Okay, so this is the first time I'm seeing the word antigenome, so let me go ahead and figure out what that's supposed to be. What is an antigenome? It's hurting my words per minute, the antigenome. The antigenome is the complementary strand of RNA from which the genome of viruses is constructed. But I thought this was a negative sense single-stranded virus. Why does it also have its complement? Do I not know what class measles are in? That's kind of concerning for me. Antigenome, yeah. Yeah, it's the complementary set of RNA. I didn't realize that measles had both. Yeah, because it says in the abstract that measles is negative stranded. So I'm going to make a note. So I'm going to open a notepad file and make a note to myself to double check that about this paper. Is measles really negative stranded? Does it have an antigenome? So weird. I weren't worry too much as you're still crushing it. Thank you, Astral. Okay, so this virus packs their RNA inside a protein package, so it blocks them from getting sliced up by siRNA, or inhibited by siRNA, I should say. Okay, so yeah, I'm gonna have to double check the genome antigenome thing because that is not something that I was aware that viruses could do. Wait, what is an antigenome? So if you are a single-stranded RNA virus, then, you know, so DNA is double-stranded, right? But RNA can either be single-stranded or double-stranded. From my understanding, I thought measles only had a single-stranded RNA genome, but it apparently has its single-stranded RNA genome and the antigenome, the genome that is paired up with that strand. So it's not as cool as as the name makes it out to be. It's just the matching piece for the RNA genome. Yeah. Nuclease attacks or high salt concentrations, as recently shown by the crystal structure of short vesicular stomatitis virus and rabies virus nucleocapsids, which, hey look, rabies, which both pointed out that the RNA is fully embedded within the nucleoprotein oligomer. Okay, all of this is saying that the measles virus packs its RNA into a protein capsule, which is adaptive, uses it to shield the precious genetic cargo from destruction in the cell. What role does interferon play in an immune response? So it is the first response that a cell can have, and it does all sorts of crazy things like shut down protein production, hold on to viruses and not let them leave, tell other cells to increase their antiviral responses, basically essentially making the cellular environment as hostile as possible to a replicating virus. Okay. Viral genome is used as a template for transcription of the unencapsulated RNAs and replication of positive stranded antigenome. the complementary template for genome replication. Ah, here's that word again. Antigenome. Viral genome is used as a template for transcription of unencapsulated RNAs and replication of the positive stranded antigenome. Complementary. Okay, figure S1. So already there's a supplement here. Maybe that supplement has a helpful picture, because I'm not... It does have a helpful picture. Okay. Okay. Schematic view of measles virus and recognition by RIG1. So I will show this. I do have to download it though, because I don't think it's in my PDF, right? I'm gonna scroll real quick. Let's go all the way down. Yeah, it's supplements not included here, which is super annoying. So I'm gonna go ahead and download that figure, and then I'll pop it on the screen just so that we can get a sense for what this is referring to. So go ahead, OBS, and add an image source. Antigenome. And this is also going to allow us to see an overall schematic view of what it is we're supposed to be taking away from this first part. Because I'm already pretty shooketh, right? I'm immediately kind of confused, because I guess I don't know how viruses work quite as well as I thought I did. So if we take a look at this, this is the schematic that they have, the measles virus RNAs and recognition by RIG1. So on top you have the genome, and so that is the schematic view of just like these are the genes that are encoded by the measles RNA, and then here, down here, we have its complements. So it is the other strand of RNA. And I was not really aware that there was an antigenome. Yeah, I'm going to have to double check that and learn a lot more about the basic biology of measles before getting too deep into it. Although, if we have any virus experts, that you could help out a lot here. Okay, let's go ahead and hide that. To make more of the strand, you first have to make the positive strand, which then serves as a template. Oh, maybe I just got it confused. I thought negative stranded viruses were ready to be made into protein immediately. Is it the other way around? I always get this confused. There's a lot of dichotomy in biology. Okay, let me not make a fool out of myself any further and look this up. Negative sense RNA. I always get it mixed up. Synthesized. Ah, they have genomes that act as complementary strands. Yes. Lunkil is right. Thank you. Lunkil again saves the stream. I always have such a hard time remembering which one is which, but yes. Okay, so Men in Black, forget Flash. Okay, let me redo a little bit of that introduction. The biology of non-segmented negative strand viruses, hardly compatible with the production of such dsRNA. This is why I thought that this type of virus was ready to go right away. But apparently this first has to be made, the antigenome first has to be made, which means that there is like a double-stranded RNA template, right? Synthesized, yeah. So I don't really know what this is about. I think this is kind of an interesting assumption that I made that I don't quite understand here. Man, I need more soup for this. I maybe I do too, because again, I think I don't understand this basic virology as well as I thought I did. Because yeah, this abstract is implying that non-segmented negative strand viruses don't produce double-stranded RNA, but in order to make the antigenome, you will have a double-stranded intermediate at some point. So that's confusing, and I'm definitely going to have to actually look that up later, but we're not going to worry too much about it. I think Local thankfully helped right the ship and get me back on track with the idea that before measles can do anything, it needs to take its single-stranded RNA genome, make the complement to it first, and then that complement, that antigenome, gets made into protein. Yeah, I'm I really wonder what the authors meant by double-stranded RNA is usually not present, because I mean, yeah, when you make the anti-genome, it has to be present at least for a little bit. Yeah, non-segmented, hardly compatible with the production. Yeah, I don't know. This is a weird sentence and I don't understand it, and it's maybe my fault, or maybe it's their fault. I don't really know yet. But we'll figure it out as we go. Okay. Viral genome is used as a template for transcription of unencapsulated RNAs and replication of positive stranded antigenome, notably complementary template for genome replication. Okay. Notably, the nascent genome and antigenome are concomitantly encapsulated, and hence viral complementary RNA strands are poorly prone to anneal. Anneal to what? I think we are running into a language barrier. I think this does not really quite read as very fluid. Okay, so the regular measles RNA genome, and when it copies its genome in order to be made into protein, are both encapsulated inside a protein structure. Hence, viral complementary RNA strands are poorly prone to anneal. Okay, dude, I don't Uncle, if you're around, I'm suffering. I'm actually suffering here, because by definition, when you make the antigenome, it am I great? Am I Why would it be poorly prone to anneal? If they're complementary, they should be binding. Why is it hard to anneal? I don't get it. I am so actually confused, and I really wasn't expecting to be this confused this quickly. But okay, we'll see if we can work backwards and try to solve whatever this issue is. Like, I'm worried I have a very deep misunderstanding of how this is working. I'm pretty sure they mean both strands are immediately bound by proteins that prevent them to bonding with each other. Oh my, thank goodness you're here. Because I don't know if I would have been able to put that together by myself. Okay, so with that context, the nascent genome and antigenome, I'm so thankful you're around, man. They are both packaged into this little protein shell and are likely bound to the protein before they can bind to each other. Okay, that's not really what the sentence says, but I think that's the only way that it makes sense. So look, I think I agree, and hopefully we find evidence for that soon, because I'm just so confused right now. Indeed, no detectable amounts of double-stranded RNA have been found in the infected cells. We therefore search for the pathogen-associated molecular pattern and the pathogen receptor involved in the cellular detection of infections by the measles virus. Okay. Whoa, okay, that's tough. Okay. So to summarize, measles first has to convert its single-stranded RNA into the complementary form of its RNA in order to be made into protein. This happens in a little protein shell that protects it from attack by the host cell. So that makes it kind of difficult to understand what is it that the cell detects when it's responding to the measles infection. So that, I believe, is what I am getting out of this paper so far. That is the problem that is set up. Yeah, okay, so let's see the results. A virus transcript acts as the pathogen-associated molecular pattern. So this title is saying that a messenger RNA is the thing that the cell detects. Okay, but this is also kind of crazy, because, hold up, since the expression of every individual viral mRNA failed to activate the interferon beta gene, this is already saying that there's a transcript involved in okay, maybe by transcript they are referring only to mRNA and not to whatever this leader RNA is. I'm getting tilted. I'm getting tilted because I'm confused. Okay, a virus transcript acts as the signal. Okay, Sure, that's not what I'm led to believe from the abstract, but okay, I accept this and I will move on and see if it makes more sense as we continue. Okay. An active viral polymerase is necessary for the induction of interferon response by the measles virus, and there's a figure. To identify which of the two activities, transcription or replication, is required for the production of the signal, we'll just call PAMP the signal, we compared the kinetics of accumulation of interferon beta mRNA with the rate of measles virus transcription and replication using RT-qPCR assays. We'll talk about that in a bit. Interferon beta mRNA accumulates exponentially in a manner that is paralleled by that of the viral N mRNA, well ahead of the accumulation of the genomes/antigenome. Whoa, okay. Okay, so this is saying that the host response, right? Host is making interferon mRNA. This happens much faster, or this happens at the same rate as viral transcription, ahead of the genome/antigenome. Okay. So there are two processes going on. Viral N mRNA. This is indicative of the virus making copies of itself, its own proteins, well ahead of the accumulation of its genome/antigenome. So this is okay. Now I'm confused again. The accumulation of genome/antigenome is delayed for the first 15 hours until a cytoplasmic N protein concentration triggers activation of replication. Oof. Reciprocally, the progressive inactivation of viral genome by increasing doses of UV applied to the virus before infection results in a parallel decrease of interferon beta and the N transcription, whereas the replication process is blocked already at the lowest UV energy. When UV energy exceeds 2000 ergs, okay, both viral and interferon, I am so confused. Both viral transcriptions are abolished. I am so lost right now. Holy cow. I don't think I've ever been this lost reading a paper in a long time. Okay. This is content, I guess. You know, it's not all sunshine and rainbows. Sometimes it's really hard to read papers. What the heck is this? Okay. The accumulation of genome/antigenome is delayed for the first 15 hours. But, okay. Oh, what does that mean? Because we see that the viral mRNA is accumulating instantly, pretty much, upon infection, which means that the process by which the measles infection is happening is producing something that is an active infection, but that's somehow before the genomes replicate. Parallel, that means the mRNA is also being amplified, but I thought measles couldn't be amplified without being converted into an antigenome first. I guess what this is saying is that the antigenome is made, but the amplification of the antigenome is faster than the replication of the antigenome. Okay, I don't know if this is making sense to anybody, but essentially what I think this is saying is that, okay, you need to convert the there has to be both the genome and the antigenome in order to make productive viruses. And I think the rate at which this requirement happens is very slow, but it is fast enough that you could get an exponential increase in viral mRNA production. Okay, I think that makes sense to me. It might not make sense to you because I may have butchered the explanation, but I'm just talking out loud to make sure that down the line I can maybe hope to explain it better because I'm deciphering this live. This is tough. This is tough. Very, very tough. They're duplicating themselves and making protein simultaneously. One process is slower, methinks. Yes, exactly. That is what this is saying, Dreamless. It's just the process by which the mRNA is made requires there to be amplification of the antigenome. But as you were saying, yeah, that's the nuance that I didn't quite register with me, is that you don't need a lot of antigenome to have a lot of mRNA. And I think that's what this is trying to draw attention to. Do you think the authors will be available on a very, very short notice to explain it to you? I mean, maybe if I were Mr. Beast or something, if I had a little more pull. I need Fi to keep on getting confused to nerf his score in the words per minute spreadsheets. No, I need to keep my spot on the leaderboard. This paper is gonna seriously hurt my average number for sure. Okay, but I think that is how that happens. And then they're talking about something about UV, so we'll interpret that now. Reciprocally, the progressive inactivation of the viral genome by increasing doses of UV applied to virus before infection results in a parallel decrease of interferon beta and N transcription, whereas the replication process is blocked already at the lowest UV energy. Okay, so one more time. Before infection? Okay, so if you break the RNA within the measles virus, that also breaks the interferon response and also breaks the ability for the virus to make protein. Okay, that makes sense to me. So when I'm cooking, chat. When you break the genetic material inside the measles virus, the cells no longer react, which means that whatever the cell is detecting to fight the measles virus is locked up in something that is UV sensitive. So we conclude that it's genetic material because we know that it matters, but it doesn't have to be. This is, being very careful with interpretation. But this is evidence to say that the actual thing, the pathogen-associated molecular pattern, the thing that the cell is detecting when it's detecting a measles infection, is in fact genetic material. Because when you irradiate the virus, all the proteins are intact, right? It's in a protein capsule that is unharmed. So it looks like a virus. It's just that its DNA, its RNA has been screwed up. And when you screw up its RNA, the cells don't really care anymore. They don't make that interferon beta. Okay, let's go. That makes sense. With that in mind, let's take a look at the data. So figure 1, interferon beta activation correlates with measles virus transcription and viral derived leader, but not viral mRNA. So we'll figure out what that means later, but first we'll take a look at figures A and B. So let's see here. mRNA copies per microgram of RNA. So this is a measure of the RNA response. So here we have interferon beta. This is the host cell reacting. So this is hours post-infection. So at time 0, you take your human cell line, you dump the measles virus on, and then you measure the amount of RNA over time. So it looks like at about 10 hours post-infection, you see a massive spike in the cell response. This is happening at the same time as the N protein. So this is the viral transcript. This is the instructions to make the N protein that the virus is pumping out. But then there's a delay when it comes to the replication of the genome plus antigenome. Yeah, so it looks like that doesn't get started until about 25 hours post-infection. So this is kind of bizarre to me because I don't know when this antigenome is made in the measles virus. I might have to alt-tab and Google that real quick. Measles antigenome. What is up with that? Because I thought it was single-stranded. Does it have antigenomes in itself? In which case, is it two single-stranded? Oh, is that why I'm getting so confused? Are there viruses with both a genome and an antigenome that are not bound together in its capsid? This is probably something that's very infuriating to virologists because I'm not like a formal virologist. That's very bizarre. Hello, I am now a free man. Welcome, Yan Yan. From what I can gather, the positive stranded RNA that's used for transcription and the positive stranded RNA that's used for making more are entirely separate. The antigen. Okay. Okay. Yeah. Okay, so that is weird, right? That's weird. That's why this is so confusing, because this is weird. So it has two sets of RNA, is that right? Hence why we can separate transcription and replication. Okay. Oh my gosh. It's so I would be so I would be struggling if you weren't here. Okay, so I'm gonna double check that though. So I believe you, Lunkill, but in order to make my video, I'm gonna double check that. And in my notepad, I will write, does measles have more than one set of genomes? Because I don't recall reading that in that might be something you just have to know. This is the struggle of learning any new virus, by the way, or any new organism, is there are just all sorts of assumptions that are made. You know what? Maybe it's time to look up what a leader mRNA is. Leader RNA. What is this? What is this thing? Leader sequence of retroviruses. Okay, what is a leader sequence? Come on, just give me the article. Why do you need me to log in? Let me at the information. What is a leader sequence? The region of a messenger RNA molecule that precedes the coding sequence of a gene is called the leader sequence. This is also, okay, I know, but is that the same thing that this paper is referring to? Holy cow, dude, I'm so gone. We postulated the involvement of the leader RNA, which is a small not capped, firstly transcribed. Okay, so that doesn't actually have to do anything. This is just the part before the RNA that's used to make protein. There's an additional bit called the leader sequence that is recognized. Okay, so that doesn't really explain the whole double set of RNA thing, but I will get into that at some point to double check, because this is very eye-opening. Yeah. I'm also very confused by what this XP stuff is. Yeah, I'm not really getting it. But what I can tell, okay, the thing that I can definitely understand here is that there's something in the RNA of the measles virus that is the actual trigger. Because when you blast the virus before infection with UV, when you ruin the viral when you ruin the viral RNA but not ruin the viral protein, the cell doesn't react anymore. So that, I think we can hang our hat on that particular fact here. Can we just defeat the authors and comment and absorb their knowledge? I will phagocytose them. I will phagocytose them and display their antigens to teach myself what any of this actually means. Okay, so panel D is not aligned with the end of panel A. This is kind of a visual annoyance, but that's fine. And then, okay, so we'll read more about these panels in the future, I believe. None of the individual capped and polyadenylated viral mRNA per se contain any molecular motif recognized as a PAMP, since their transient expression in 293Ts do not lead to the induction of any interferon beta signal 1C. Okay, so that's this. So 293T, this is a very common bog standard mammalian cell culture. This is, what is this? I don't remember exactly what 239Ts are. There's some mammal thing. What is it? It's a human cell line, but what cells are you? This is actually kind of important. Is it kidney? It's kidney, right? Heck, human embryonic kidney. Okay. This is actually kind of important for their argument, considering, yes, this is a human cell. However, it's not exactly the most representative cell. Measles is a human-only virus, but it is much more known to be an upper respiratory tract infector and an immune cell infector. So the fact that they use HEK cells here kind of not great. I would say that maybe if you could get an immortalized lung line, that might be a little more representative. So this is a result that I'd have to interpret with caution. So what is this? So this is the cellular response, interferon beta. And then here are all the different conditions. So we have mock infection. So a mock infection is you do everything the same except put the virus onto the cell. And we can see that, you know, the cells have some kind of baseline level of interferon that doesn't really react. When you give the, is this protein or is this RNA? Let's see. Lack of transfected with expression vectors. Okay, so when you put in the RNA for the N protein, there's no reaction to, there's no interferon response. P protein, no reaction to interferon response. MFH, GFP might be a control, just a foreign but non-viral mRNA. What is N.T? What is N.T? Control T7 RNA polymerase. Is that what that is? Or protein with 200 nanogram of poly-IC. So T7, so this is from a virus, but not a virus that productively infects humans. I don't know what NT is. What is this control? What is this control and why is it not in the caption? Guys! Guys! N.T. Great, no matches. Love that. No, man. What? I'm just supposed to know what N.T stands for? Come on. That's super annoying. By suffering life. No transfection. I assume this is no transfection, but why is no transfection different than mock? Mock is supposed to be no transfection, so I don't know what NT is. I love it when people don't explain the abbreviations. It's so annoying. Let's see if it's in the body of the text somewhere. This also rules out. Great, no, that's it. That's the only section that this figure is talked about. So this is some mystery control. I assume it's no transcription, in which case I don't know how their mock was done. That's probably in the methods. Mock transfection is done with oligofectamine alone. Okay. But why is that different than no transcription or no transfection? I don't try to pick bad papers, by the way. I genuinely don't enjoy like malding at I genuinely don't enjoy malding at papers. I want to pick ones that I like because they're more fun to read for me. I don't want to stress, but it's happening. It's happening. That's so bad. That's so annoying. For the record, not explaining abbreviations is against APA style. It's entertaining if nothing else. I know. It's still it's content, right? It's content, and you can't. Not every paper you read is gonna be a rock star paper, right? That's just reality. But like, I think why I'm hurt is because I love PLOS as a journal, and this paper is hurting me now. So poly-IC, I believe this is generic viral genetic material that's commonly used as a positive control. So this is, I don't remember exactly what poly-IC is. I do believe it comes from a virus, and I do believe that it is known to cause a very strong interferon response. So to make sure that their experiment actually works the way they think they do, they need negative controls like, you know, reagent alone and no virus, but they also need positive controls. Like, we know that the cell is going to freak out when it sees poly-IC. And then PBS, this is just salt water. This is another control. Okay. I like that they did so many controls, right? GFP, foreign protein that's non-viral, whatever the heck this is. T7, which is a viral thing, but not a virus that infects humans. T7 is from phage, so it is a virus that infects bacteria. Poly-IC for the positive control, love that. PBS is salt water. Wait, actually, never mind. This is PB. I think this is maybe plasmid. It just happens to have the same acronym as what is it? What is P? Phosphate buffered saline, but this P is lowercase, which indicates that this is actually a plasmid called BS. I believe that is what that is, and please tell me you define that somewhere. Oh, dude, no way, dude, no way, no way, no way, no way, man. Oh, no way. Proofread your thing. Proofread your thing. Or you're right, because conventionally, if this is phosphate buffered saline, this should be all in capitalized. If this is a plasmid called BS, then this is lowercase p BS. The only mention of PBS in the body of the paper and not in the supplemental is phosphate buffered saline, the salt water they use to wash the cells, which either means this actually is phosphate buffered saline and this first P has to be capitalized, or this is a plasmid that is not mentioned in the body of the paper. Either way, this sucks, dude. This sucks. Please, what is happening, man? How many times does PLOS need to hurt you before you realize? No, don't say that. Don't say that. I love PLOS. They might be kind of hard to read, but I think like the other PLOS papers that I've read were hard to read because it doesn't appear that PLOS has a cultural history of good storytelling, which is fine, right? It's a very like data first kind of paper, but this is not just no story. This is like mistakes, right? No definition of NT, no definition of PBS, or if there is a definition of PBS, this is lowercase. Like this is some rookie stuff, man. Like I don't know. This is aggravating. Otherwise, it's a cool figure, right? This shows that the cell is not responding to an actual mRNA, which is really exciting, right? Like underneath how angry this figure makes me is something that is genuinely interesting. Nothing that the measles virus makes on the way to making protein is the thing that triggers the cell, which indicates that there is some piece of RNA that is deactivated by UV radiation that does not get made into protein. And that is probably what the signal for infection detection actually is. Synthetic. Oh, interesting. Poly-IC synthetic. Okay. It's from 2007, right? Probably very early on in PLOS's history, and therefore quality control might not have been as strong. I'm actually unaware of how long PLOS has been around. If so, that makes a lot of sense to me. Man, we got a lecture on making figures, and I thought this is all common sense. What's the point of this? And now I see this. Yes, your teachers are there to teach you best practices because this is so... Here's the thing, right? I might be making a mountain out of a molehill with this, but when you are writing for scientists, anything that you do to put them in a bad mood is going to look bad on you, right? Like, I was excited to read this paper, as you should be. Only very, very immature, less experienced people or people with weird chips on their shoulders go in to read a paper giddy at the prospect of it being bad. I think, from my perspective. Now, this is like, you know, if you're making content, then it's a different story, right? Then you have a different incentive. But if you are a practicing scientist and you like get giddy from bad papers, I think that's wrong. That's bad. Nobody goes in hoping a paper will be bad. And I don't think this paper is bad, right? I think the conclusion is genuinely interesting. So far, the science seems to check out, right? Like all of the controls look good. I like this experiment. I think it's very cool that they thought to do this. From a scientific perspective, I don't have any issues with this so far, but you are putting me in a bad mood. I'm going to be meaner to you because you put me in a bad mood. Don't do this. Don't do this. That's so sad. NT might be no template. It's essentially a representation of background signal IDK. No template. But again, why is that different than mock? No transcript? Maybe empty plasmid? Maybe empty plasmid? So plasmid that doesn't contain anything? That's If that is the case, then that should be defined as well. There's just like no indication that that's what that is. There's people screaming about not explaining your acronyms in the lab too, if it makes you feel better. Yes, the validation. If you have an acronym, you must define it. It is insane to just drop N.T. without any acknowledgement in the figure caption. That is absolutely bonkers. Apparently PLOS One was launched in 2006. Wow, younger than I expected. Paper's already hard to read, but if paper's not helping, it's irritating. It super is. It super is. And I have to, I'm not, okay, so I don't blame the authors, okay? I think this is an unfortunate reality that, no, I do, right? Because this isn't a language barrier thing. I was gonna let them off with the language barrier thing because this is not a paper that came from a primarily English-speaking country. But the choice to not define an acronym is universal. That doesn't matter whether or not you're writing in English or not. That is a choice that you would have done in your native language as well, and that's super, super infuriating. Okay, but to bring it back, to calm it down a little bit, my righteous fury, the point of this is that combined with the UV results, these UV results, the kidney cell, right? It's not the respiratory cell, which it is in a human, or the immune cell. This is an artificial kind of system because it's not how we typically think of measles as working. We have to keep that in mind so we don't give the authors too much credit. And again, I'm in a bad mood, I've been put in a bad mood by the lack of care in the writing. And so I am going to be a little harsher, right? This is not a cell type that measles is known to infect productively. This is a kidney line. I would like to see it either be an immune line or something that is a respiratory line. Maybe they do it eventually. Maybe there are downstream experiments that validate this that are more close to biological reality. But like the sociology of reading science, right, is that now I'm a little extra suspicious because of the mentality that I have going in, right? Like we like to say that scientists are objective, that we are just looking at the facts, but that's never true, right? You are in a mindset when you are interpreting information, and my mindset has been a little soured by this. Where was I? So in this not totally reliable cell line, because it is respiratory or immune, we see that none of the measles RNAs that become proteins trigger this cellular response. So ruling out those RNAs, there must be some other RNA that is not making a protein that is the thing that the cell is responding to. And in the case of this paper, that is going to be what they are calling the leader sequence, a piece of RNA that is before the part of the RNA that becomes the protein, if that makes sense. I'll draw a picture of it, I think, in a little bit, as it becomes relevant. Yeah. From my own experience, it's probably a sheer number of revisions it went through. They probably did define them at one point and deleted them by accident. I mean, maybe. That is the kind gracious interpretation, other than just it being messy. Okay. Since they were honest here, right? Their transient expression in 293T, they didn't hide this by saying mammalian cell line, right? They were honest. This is not the typical cell measles would infect. Do not lead to the induction of interferon beta signal. This also rules out that any viral protein is responsible for interferon beta activation, in agreement with a previous report showing that protein synthesis is not required. So this is also good to hear, right? If the previous literature showed that you don't require active protein synthesis for the cell to respond to the measles virus, this is consistent with that hypothesis, or consistent with that observation, rather. The fact that you can dump the shell of the measles virus into the kidney cells and the kidney cells don't care, this is consistent with that previous report that you don't need active production of viruses for this immune response. Okay, so it's not an RNA that becomes protein. So what is it? They are going to make the argument that it is viral leader RNA. Viral leader RNA. Okay, and this activates the interferon beta response. Oh, my words per minute! It's like half the cancer paper. We gotta go. I got sidetracked in my righteous fury. We hypothesized that the leader RNA may host a signal, pathogen signal feature. Leader is the first transcript from the very 3' end of the genome and is 56 nucleobases long, not capped, and not translated. This first transcript could fulfill the requirements of the rapid induction of the interferon system after infection, as shown by the quick phosphorylation of I-kappa beta, one of the upstream signals of the interferon beta gene activation. I don't actually know anything about I-kappa beta, so I'll have to read more about that later, which is observed as early as 5 minutes after viral entry, and by the NF-kappa B DNA binding activity detected in 1 hour. So NF-kappa B, this is an immune pathway that often leads to apoptosis, so another technique that cells can use to shut down viral replication. Yeah. To be fair, half the cancer paper is solidly second place. there's a thing that a chemistry professor of mine used to say all the time, and is that there is no second place in science. There is no second place in science. Super toxic, but I do kind of respect the hustle. Okay, so let's just reread that. The leader is the first transcript. So this is a part of the RNAs that become proteins. It is not capped, so it is not modulated in any kind of way, and not translated. So this is an RNA that is not made into protein. This first transcript could fulfill the requirements of the rapid induction. Yeah, so it's basically saying, well, we destroyed the RNA and the cells stopped responding to measles. We put in every individual measles RNA transcript that does become protein, and each single one of those don't elicit a response. What's left? What's left is this weird RNA that is attached to these other RNAs that does not get made into protein. I actually don't know what the function of the leader peptide is, the leader sequence is. Generally, it's to protect the RNA from degradation. So I'll have to look up leader RNA function in viruses specifically, because there are leader sequences in regular eukaryotic RNA too that prevent those RNAs from getting chewed up. So I'm wondering if that's the case for this RNA as well. Yeah. Let's see. Myelin kind of being a kind of cryptid while Phi is transcribing HK cells as we speak. A reversal of our usual. Very true, very true. I-kappa beta is the inhibitor of NF-kappa beta. When it's phosphorylated, it stops binding NF-kappa beta and lets it do its stuff. Oh, interesting. Okay, so it is de-repressed. So this phosphorylation of this de-represses that. Okay, so that connection I understand. The in vitro transcription of measles is hardly efficient. Okay, that's actually an interesting thing that I will make a note on. Measles transcription is not efficient. Also, is hardly efficient. This is very weird sounding. Not to pick on anybody whose second language is English, but there are word choices that I think make it more obvious that that does not read as cleanly in English as a native speaker might write in. Therefore, we chose. By the way, I respect if English is your second language or third or fourth. I respect that immensely. I dislike English. It is my primary language. It sucks. So if you are here and this is not your first language, dude, so much respect for you. Therefore, we first chose to produce in vitro the leader RNA from permeabilized VSV virons, since it is biochemically indistinguishable from that isolated from VSV infected cells. Okay, sure. So we made the leader RNA. We chose from VSV. Quickly looking up that acronym. Tons of acronyms for biology. Okay, vesicular stomatitis virus. So I think this is a very closely related virus to measles. or at least I hear it mentioned. Oh, it's actually related to rabies. Why are we using VSV? This is something that might be a virology thing that doesn't need explaining to experts, but certainly to me, I'm a little confused as to why we're using that. But okay, it's not a huge deal, I think. Upon transfection, a VSV leader preparation stimulates the gene transcription, whereas the interferon response is abolished when a sample is pre-treated with RNase A. So yeah, this is same thing. You break up RNA and you don't get the interferon response. Okay, so when we make the leader, you get interferon beta. When we break it with RNase A, we don't get it anymore. So it's probably the leader that is causing the immune response. Thus, a mononega virales leader RNA synthesized by the viral polymerase can act as a PAMP to demonstrate that the measles virus leader can induce an interferon response. Okay. Maybe they used an existing system in the VSV. Yeah. Okay, again, this is like the writing thing, right? So the measles leader transcript is 56 nucleotides long, and the VSV leader sequence is 47 nucleotides long. They are implying that these are different, but I thought they were making the measles leader peptide, measles leader sequence in the VSV virus. But that's not the case. These are two leaders of slightly different lengths. This is a proof of principle. First, we will demonstrate that this happens in VSV. Then we will try to make it work in measles for some reason. I think that this is... it's motivated because measles is hard to work with. So instead of starting with measles because measles is difficult to work with, we first chose to do it in VSV. Okay, I think that this is mostly my fault for not connecting these two sentences, but I think it could have been made a little more clear. There could have been a connecting sentence or something, but I think that was mostly me misreading this. Yeah. Upon transfection, blah, blah, blah. Okay, thus can okay, this is a bit of misleading writing. Thus, a measles leader RNA synthesized by the viral polymerase can act as a PAMP. I mean, this is a very like English as a first language kind of situation, right? Like can is technically okay to mention here, but I think for me, I would rather say potentially or may act as a PAMP, because it sounds a little more affirmative than it is. I don't think they are meaning to argue that they have proven that measles leader RNA can act as a pathogen signal. And I think it's okay that they use the word can here, but better, I think, potentially works to make that a little more clear. To demonstrate that the measles virus leader can induce an interferon response, a leader like RNA was transcribed in vitro by the T7 phage-dependent RNA polymerase from an engineered plasmid. Okay, so because measles sucks to work with, we are going to make the leader sequence in an artificial way by using T7 RNA polymerase. So why does this matter? This matters because you are replacing some of the machinery that measles uses in order to make the same sequence. So why this matters is that maybe it's not just the leader itself, but the leader bound to a measles protein, like the combination of RNA and protein that is being detected. So I think this is actually pretty responsible, right? This is saying that we would love to do it naturally just with the measles virus, but it sucks. So we artificially changed the system to test this one aspect. Is it just the RNA leader sequence? The other thing about replacing the measles machinery is that you're also replacing whatever consequences those machinery had on the RNA chemistry, right? So maybe the measles virus proteins are chemically modifying the leader sequence, and that's very important for the detection inside the cell. So they're communicating that there are limitations here, which I do appreciate, right? Like I'm not gonna hold a grudge against the authors forever. I am trying to be as objective as possible here. The difference between can and may is very basic, if not omnipresent in all languages. From the point of a non-native English speaker, it's a very embarrassing mistake to make, if it was a mistake. I am willing to believe that it is a mistake because, you know, I'm so willing to forgive non-native English speakers of these kinds of mistakes, just because like I'm dog water at my second language, right? Like I can't even imagine, like so many students want to study in, I should say, historically, many students want to study in the US. And to me, having worked with brilliant graduate students from across the world, dude, I couldn't do what they do. They're much brighter, more brilliant than I would ever be. And on top of that, they also learned a whole goddamn new language. Right? My second language is garbage. I'm really bad at it. It's very embarrassing to speak my second language, so I'm very forgiving of that kind of. I'm not going to call it a mistake, but I'm very forgiving of that choice, I should say. This single-strand leader RNA turned out to be a strong inducer of the interferon beta transcription in these kidney cells, with the PAMP activity being destroyed after RNase treatment. So this is kind of repetitive too, but I appreciate the reminder. So figure 2a. So interferon beta mRNA, so this is the cell response. Okay, bunch of stuff here. And then we have the poly-IC. So what is this? Let's go look at the caption. In vitro T7 transcripts independently of RNA, purified according to their size. Short single-strand leader. Okay, short single-strand leader. So okay, so I guess this is mock. This is LE's leader. D-leader, an anagram for leader with poorly predicted structure. Okay. I'm sure that'll be explained later. Single-strand 86 nucleotide MIC RNA. So MIC, if it's eukaryotic MIC, right? Because MIC is a eukaryotic gene, unless there's a viral MIC as well. I'm gonna have to look these up later. Short hairpin EGFP or poly-IC. Okay, we probably have to look deeper into why they chose these, because it's not entirely clear. Okay, MO equals mock. See, they defined other things. They just forgot that time, which is frustrating, because how do you not comb over this repeatedly before you publish? I guess back then, if PLOS's reputation wasn't that good, maybe they just didn't care. As controls, RNAs were digested with RNase before transfection. That's not really a control. That's kind of your whole experiment. Okay, but what we can interpret is this part. Okay, so when you give a mock infection, you don't infect with anything. You get a baseline level of interferon. When you add in the leader sequence, you get a huge spike in interferon beta in these kidney cells. That makes a lot of sense. When you destroy that leader sequence, it's all the way down here underneath mock, kind of. And then the rest of these I don't really care too much about. I don't even really understand it at this point, but that's okay. What I got out of this, what is needed, is that you add that leader sequence that doesn't code for protein, and the cell freaks out. You do the same thing, but you destroy it first, and the cell stops responding. So it is dependent on the leader sequence. Yeah. Doing science and writing science in a second language is insane to me. Yeah, exactly, man. Like, dude, mad respect, mad respect. Now, I don't think there is any mistake with the can. They're talking about the common order that both VSV and measles belongs to. If they, I mean, for me, I wouldn't use can just because they share the same family doesn't necessarily that measles automatically that's automatically true for measles. So with the VSV, they're showing that something from that order can indeed work like this. Yeah, I still would replace the word can with potentially, because it's less ambiguous what you mean by that. Plus one's reputation in psych when I started grad school there was pretty bad, TBH. It's gotten a lot better in the recent years. Yeah. As long as I've been reading research, Plus has generally been pretty good. Or maybe I haven't been paying enough attention. I don't really know. Or at least when I started caring. Where I don't know about when I started reading Plus. I know that when I started caring about journal reputation, Plus has always been seen as pretty good. Yeah. Okay. Thus, this leader RNA synthesized by the viral polymerase can act as a signal to demonstrate that the measles virus can act as an interferon response. A leader-like was transcribed. Okay, we already got that. Great. That makes sense. They didn't say it was true for measles specifically. Yeah, I that's why I say that I don't think it was wrong to use the word can. I think it is a preference to prevent confusion. I'll say that to prevent, because when you replace this word with potentially, there's unambiguously no way you can confuse what they are saying there. Yeah. Again, I think it's fine. It's technically correct, right? I'm not saying they're wrong here. It's technically correct. I just would use a different word. Okay. So, what do we have next here? 5' triphosphate end is required for optimal activation of interferon beta. So 5' triphosphate. So this is one end of the RNA that the measles makes. And so it is saying that when you add that triphosphate group to that end of the molecule, that makes the cell go crazy. Optimal activation of interferon beta. So it's not just that the leader sequence is important, is that it has to be chemically modified in a way that the host can detect. Okay, so measles and VSV leaders share a common 5' triphosphate structure that could account for interferon activation. Indeed, the role of this end of the transcripts can act as an induced RNA. Okay, did I disconnect from OBS? Are we back? We chilling? I think we're back. OBS kind of freaked out a little bit, but I think we're still live, right? Please let me know. Or I guess if people are just generally silent, I'll know that something is horribly wrong. But I think I'm okay here. So okay, great. Yeah, OBS was like, we lost connection. Okay, but we're back. Cool. Okay. Using different T7 transcripts, including a deleter, which has the same basis as the leader, but in a scrambled order, a mic tag. Okay, so here is where they're explaining the rest of that figure, which I appreciate. I knew I was going to learn about these eventually. I don't think they would have left all of those controls out. Using different T7 transcripts. Okay, so this is taking the exact sequence of the leader, but mixing up the order. I actually wrote a little program to do this automatically, which was kind of fun, in a way that was ensuring sufficient scrambling. That was one of my graduate school projects. What do I mean by sufficient scrambling? I wanted to scramble everything about the protein I was working with. That included substructures as well. So to make sure that those substructures did not accidentally reappear. So I made a targeted scrambler, I should say. A MIC tag sequence. Yeah, so MIC, this is a mammalian eukaryotic kind of protein. Although not really talking about why they're using a MIC tag, I assume the quiet part is that they just wanted some other protein that was mammalian, eukaryotic. And a small hairpin with the 12 base pairing of the coding sequence of EGFP. Again, not entirely sure why this part matters, and I don't think that they are going to explain it. We confirm the T7 transcripts, independently of their length, sequence, or structure, do possess PAMPs. Independent of their length, sequence, or structure, do possess PAMPs. Do possess PAMPs? I have to think about that for a second. So it makes total sense why they would scramble it, right? Because you just want to see whether or not a random string of RNA of that length matters. But in a scrambled order, a MIC tag. So that's just a different mammalian protein. Independently of the length, sequence, or structure. Confirmed independently if they do possess PAMPs. Okay, so let's look back at that figure 2A. Okay, so... Okay. Leader sequence. Cell freaks out. Makes sense. That's our hypothesis. You add in that RNase A, you destroy that RNA, it shuts up. Okay. So if you take that same leader sequence and you scramble it, you actually still get that RNA reaction. So that is really, really interesting. So my assumption was that the sequence of the leader sequence is what mattered. But this is saying that even if you randomize the order of the leader sequence, you still get an intense cellular reaction. So that is maybe something worth putting in the video. I think it'd be cool for you to see me work this out in real time and then see what it becomes in the video. That might be kind of neat for some of you. So even scrambling the leader sequence still causes an immune response. Okay, so I'm going to pause there, chat. I'm going to say that whole part again, and I want to see if I can explain it to you, and you can see if you understand it as practice for me. So what this is saying is that there's something locked up inside the measles virus. We know that it's not the protein. There's some okay, sorry, let me back up. The question this paper wants to answer is, what is the molecule that the host responds to during a measles infection? You don't need protein, or it doesn't recognize protein, because you could dump the virus, the measles virus protein on, and the cell doesn't react. When you destroy the RNA, the cells don't react. So, the molecule that the measles virus is signaling to the immune cell to cause an immune response is some molecule of RNA. We learned earlier in this paper that none of the RNAs that are used to make protein are the culprit. So whatever RNA is actually getting detected by the cell is something that does not code for protein. This is the leader sequence. Here's the cool thing that I think is really fun to put in the video, is that this doesn't appear to be sequence dependent. So the cell isn't detecting, oh, this is the leader sequence, this AGUU, CCU, CAA, whatever the leader sequence actually is. The cell doesn't care about what the code of the leader sequence is, because when you scramble the code, the cell still has an immune response. And I want to pause here to take a poll to see if that nuance got across. The A's, the U's, the C's, the G's, the order that happens doesn't appear to matter. And so WittyMI is asking, but how, why, what are PAMPs, if not length, structure, or sequence? And that's, I think, something really unexpectedly cool about this paper, right? Like, I didn't read this paper beforehand, I just read the abstract, but this is, I think, the part that I will put in the video, because this is a big mystery, right? I thought, okay, there's some sequence, some motif, some sequence of A, G's, U's, and C's that the host is detecting. But that intuition turned out to be wrong, because when you randomize the order, the cell still thinks it's getting under attack. It's still under attack by the rabies virus. I think that is that's insane, right? That's very, very interesting. So mutations in the leader sequence are detected and don't lead to immune evasion. That's nice for the host. Yeah, guys. So this maybe indicates that there's some robustness with regards to what a mutation could even do. I guess the next mystery is I don't really know what this MIC thing is. Maybe MIC is a known inducer of interferon. Is this the same MIC that I'm thinking? Maybe this is a different MIC. Anti-MIC. MIC short RNAs. You know what else is super annoying is that they don't spell out what this particular MIC actually is, because sometimes you can have acronyms overlap. So I'm gonna check to see if this is the same MIC that I'm familiar with. What protein are you? Anti-MIC protein. MIC tag. Yeah. I think so. I think this is the same MIC I'm familiar with. Surely. MIC protein, family of transcription factors. Yeah, like this is something that is often implicated in cancer. So I will be a little hard on this paper for not motivating why they chose MIC, because I don't know, maybe if you're a virus guy you don't need this explained to you, but you know, I've read my fair share of immunology, virology papers at this point. I've never really heard of MIC being used to activate the immune system, because to me MIC is more of a cell cycle cancer kind of protein, not an immune viral infection kind of protein. So I'm a little bummed, and I don't think that this gets explained, which is kind of annoying. A small hairpin with 21 base pairs of the coding sequence. We confirmed that T7 transcripts independently of their length sequence of structure do possess PAMPs. Okay, so okay. They said that T7 transcripts contain PAMPs. All of these transcripts were made by this phage polymerase. So, okay, I think this is their attempt at being honest. That this MIC tag, this is not, I don't think this is supposed to cause an immune response, but it still does, because the T7 polymerase is doing something to MIC to convert it into something that the cell recognizes as dangerous. Okay. I think that's what this is trying to say. That yeah, and I think that makes sense with the language that they use, right? We confirmed that T7 transcripts. This is not measles. This is our artificial system causes different transcripts to become PAMPs. So it makes the leader sequence into a pathogen-associated molecular pattern. It makes the scrambled version into a pathogen-associated molecular pattern. It makes this random eukaryotic protein into a pathogen-associated molecular pattern. And it makes this unusual shaped RNA, the small hairpin, combined with the coding sequence of EGFP into a pathogen-associated molecular pattern. So this is kind of saying that the system, this non-measles system that they use, potentially has an issue, or maybe not an issue. Too early to tell whether it is an issue, but it is being honest in the sense where this is a consequence of the T7. This is not a consequence of measles right now. Okay, I respect this. I think that is good to admit. And I understand now. I've reached the understanding. And if I'm right, we will see this be addressed later on into the paper. So it's basically saying that everything is a pathogen in the model. It's saying that whatever T7 did to make this RNA made them into infection signals. So as Lunkel is saying, they want to show that it's mainly about the 5' triphosphate. Yeah. So this is building up to whatever it is that this T7 polymerase is actually doing. Yeah, I got a little confused, but that is what this is saying. This artificial system is doing something to make these RNAs into a trigger for the immune system. And so I think what they're about to talk about is that the T7 is adding this 5' triphosphate molecule to make them more into something that the cell would recognize. In fact, I think that's the next sentence. I should have read one more sentence in, and I would have gotten that on my own instead of struggling. That's okay. Struggling is a very normal part of learning, and I want everybody to make sure that every video that I pump out is the result of a lot of struggle. It's a lot of reading. Can you please remind me what T7 is? T7 is a phage, a bacteriophage, a virus that infects bacteria only. And this particular phage makes an RNA polymerase that slaps. It's like, it's so good at making RNA. And so they are using this, thing that doesn't happen in measles to make their RNA because measles sucks at making their own RNA. So they're using an artificial system. Okay, so furthermore, converting the triphosphate into a monophosphate moiety at the 5' end of leader and short hairpin RNAs before transfection, either by calf alkaline phosphatase, data not shown, cold dead, could have been in the supplement, but okay, or by RNase H, digestion of a longer RNA precursor, partially annealed at the 5' end to a short complementary DNA. Oh, it's annoying that that's in the supplement too. Reduces their ability to activate the interferon response. Okay. Yes, indeed the role of the 5'. Okay, yes. Yes. Okay, so it actually was clear. This was just my misreading. So they actually do mention that T7 does this neat little thing where it adds this chemical group. And it's this little chemical group. Sorry, if you are good at biology, it must be kind of infuriating watching me digest it with faults. But yeah, as I said, I'm being honest. I don't read things correctly the first time around. But they do mention that this particular enzyme that they borrowed from phage, does add this unique chemical group onto RNA, and this unique chemical group onto RNA can make it look like they're being infected with an actual virus. And so this is saying that T7 is doing something to make RNAs into attack targets, and they hypothesize that it's this 5' chemical modification. So when they change the 5' chemical modification to, instead of a triphosphate, so 3 phosphates into a monophosphate, 1 phosphate, it looks like that gets the cells to calm down, reduces their ability to activate the interferon response. Thus, 5' triphosphate represents a pathogen-associated molecular pattern, even on short single-stranded RNAs. To mimic viral synthesis of measles viral leader in the cytoplasm, cells stably expressing cytosolic T7 phage polymerase were transfected with a plasmid, encoding the viral leader under the control of T7 promoter. As a control, the leader was alternatively put under the control of the H1 promoter, which is recognized by the cellular RNA polymerase 3. Interferon beta was significantly induced only when the viral leader was transcribed by cytosolic T7, not by the nuclear endogenous RNA polymerase. Very cool. Okay, so we're cooking now. Now things are getting interesting. Okay. And importantly, the measles leader has the 5' triphosphate and under normal conditions as well. That's not an artifact of the T7. Exactly. Those two things both have to be true. They both have to be true. Just call it PAMP. I'd like to spell it out as much as I can or replace it just in case someone is new to the stream. I try not to rely on too many of these acronyms. So if we look at C, so mRNA copies. This is the figure that we should be looking at. So mock infected. So this is cells that are either the normal kidney cells or the kidney cells that have T7 cells. Give me a second. In T7 driven, but not in vivo, Pol3 driven transcription activates different beta. Still stably or transfected with T7 leader. Okay, so in black, we have kidney cells that have been modified to make T7 polymerase. So making this enzyme that we borrowed from bacteriophage. And then here we have MOC, so no infection of any virus. In T7, we have an infection that enables the cell to make the T7 leader. So in cells that are making the correct polymerase, when you give them the correct sequence, you see a huge increase in host response. If you change out the sequence to the wrong sequence, if you change the regulation to the host polymerase, you don't see quite as much of that activation. I don't love that we don't see significance bars. That's kind of annoying. Actually, don't appear here either. Whoa, dude. I don't know if this is a thing of old PLOS or something, but hello? Significance? Was it in the other figure? Oh my gosh, I didn't even notice. No statistics. What? Bruh. Man, what a different time. That's actually kind of crazy. No, I didn't even notice this far into the paper, and I didn't even notice that there's no statistics included. That is shocking. Yeah, Mylan, avert your eyes, man. What? I mean, I'm sure that they are significant with error bars this small. But like, what? That's not in the caption either, I don't think. Is it anywhere? P equals P less than P greater than? Huh? Statistics? No. Oh my gosh. No way, dude. No way. Yeah, I just did a quick scan for the stats. I don't see them anywhere. Maybe in the body of the paper? I don't see it in the body of the paper either. Man. I mean, maybe things were different back in 2007, but ugh. Oh, that's rough. In any case, this is all pointing to the culprit. The culprit is the enzyme that copies over the measles RNA, is doing something to that RNA, and it's probably adding on a 5' triphosphate that tells the cell that it is infected. So we are narrowing down on the actual signal that the cell responds to. If I can't do this, fine, I'm gonna explode fire. I know. You could just BS your day in 2007. I'll say this, right? For as much as people love complaining about the issues in science, never lose perspective that science itself is improving over time, right? The standards for the scientific method, it do increase. And while there are journals that plop up that are schlock, right? AI slop, right? Like, of course, there are going to be issues as we progress into the future. Overall, I think the quality of science is improving on I don't know about research that shows this. I read a headline on Reddit one time showing that psychology has improved by quite a bit in terms of replicatability. I would assume motivated by the replication crisis. It looks like there's an actual substantive improvement of psych studies, and I have to imagine that's true for all fields, that the arrow of time is improving. And just to look at this paper from 2007, not report stats and get these caption typos is actually kind of crazy. Yeah, the fact that this is unthinkable now is a sign of progress. Yeah, absolutely. is this 5' triphosphate end of an mRNA different from the cap? I believe the cap is something else. I think it's a different chemical group, but I will look it up right now. mRNA cap, because I think the cap is like a series of, oh no, that's the poly A tail. What's the 5' cap? Let's see. Altered nucleotide, mRNA capping, an unusual 5' to 5' triphosphate linkage, ribose structure, capping process, terminal phosphates removed, leaving a biphosphate group. This is all chemistry. Wah. Wah. I think caps probably, there are probably several different caps that can be added. The starting point for capping with 5' methyl guanylate is unaltered 5' end of RNA molecule, which terminates as a 5' triphosphate group. This feature is a final nucleotide followed by 3 phosphate groups attached to 5' carbon. The capping process is initiated before completion of transcription designation pre-mRNA is being synthesized. Yes, so that is an excellent question. Whether or not this is in form differently is something that I have to look into. Is this, the cap is guanosine, so that's stuck on the wrong way, I think. So 5' triphosphate guanine. There's a triphosphate between the mRNA and the guanosine. In the case of the 5' triphosphate, there's no guanosine. Okay. I'm going to go ahead and just copy and paste that into my notepad, that whole conversation. Thank you, Longo, for checking on that, just to make sure that I refine my own understanding of how that works before I put it into a video. Yeah, I'm going to have to re-educate myself on the capping process. Okay, nice. It's nice that I get, because this ended up being a lot more taxing than I was expecting in terms of testing how well I understand each individual bit here. Converting to triphosphate, transfection, PAMP. Let me go back and read the abstract one more time to see if it was there, or no, the introduction. So I'm going to read to myself a bit just to really lock in for a sec. Because I don't really think that they explain too much about the basic biology of the 5' cap here and why it's different in eukaryotes. Oh, actually, I think I saw it. I think they're literally about to explain it. So we will march on. To mimic viral synthesis, stably expressing transfected as a control. Okay, we did that experiment. Likewise, the scramble transcribed in the nucleus by this polymerase did not do the interferon thing, which is understandable. It needs to have that 5' triphosphate given to it by the T7 to become quote-unquote dangerous. Transfection to silence. Sure. Okay. This is the question that I was thinking about, which is good because they're addressing it immediately. What would the in vivo relevance of the recognition of the 5' triphosphate motif in the cytosol as a danger signal? A major feature of the nuclear and mitochondrial transcription by either RNA poly 1, 2, or 3. All but one RNAs are processed at the 5' end either by cleavage or by a capping process resulting in the loss of the primary 5' triphosphate end. Major feature by either these 3 polymerases is that all but one RNAs are processed either by cleavage or by a capping process, results into the loss of a primary 5' triphosphate end. The only known exception is the abundant 7S cell RNA that is part of the signal recognition particle, but it's inaccessible as it's shielded by the P914 protein subunit. Accordingly, the cytoplasmic and nuclear RNA extracted were found to be incapable. Sure, I think that makes sense. Accordingly, cytoplasmic and nuclear RNA extracted from the non-infected T. Furthermore, cellular RNA transcription from RNA template does not occur in mammalian cells because of the lack of RNA-dependent RNA polymerases cellular homologue. Oh, cellular RNA from RNA template. Okay. Okay. 5' ended RNA in the cytoplasm thus represents a well-suited feature for a danger signal, a molecular pattern that is absent from uninfected cells, according to the concept described by Metzinger. To propose that it acts as a PAMP used by cells. Okay, I might just be confused because don't you have 5' caps on eukaryotic transcripts anyway? 5' triphosphate ended RNA in the cytoplasm thus represents a well-suited feature for. Does it lose the cap by then? I'm confused. I think they're just saying that the 5' triphosphate doesn't appear in the host RNA, but doesn't regular transcription also involve 5' capping? I know they get uncapped eventually. Where do they get capped? In eukaryotes, I'm reading the Wikipedia article for 5' cap. In eukaryotes, the 5' cap found on the 5' end of an mRNA molecule consists of a guanine nucleotide connected to mRNA via an unusual linkage. Yeah. This is more common in higher eukaryotes, recognize mRNAs from other organisms. Is it because this is linked in a way that is not the same? Is it because their 5' linkage is different than our 5' linkage? The 5' cap is more than just the triphosphate? The 5' cap is more than just the triphosphate. That is true. Okay, the 5' cap has triphosphate, but also has nucleotides. Okay, so it does look different. Chemically, they do look different. Okay. I think I got thrown off because when you shorthand 5' cap, it's usually in the context of the nucleotide, the phosphates. Like, I just didn't remember completely the difference in architecture. Okay, so if I'm understanding this correctly, in eukaryotes, the normal mRNA is processed with something called the 5' cap, which, yes, does contain triphosphates, but also contains altered nucleotides. So the hat that the regular mRNA uses is different than the hat that the measles virus uses, even though they share the same components. So, like, the measles hat is maybe like what could I say here? The measles transcript is like a headband for the mRNA, but then the normal host mRNA is like a headband but with cat ears on or something like that. You can kind of get a sense for like me writing out the visuals live. Please let me know if that is what that is saying exactly. Thank you so much. Because I think the paper kind of assumes that people remember a lot about 5' capping, which is maybe fine. Like DNA stuff was like mRNA and DNA stuff was always my weak point. I've made that known quite a lot on stream. That, you know, in terms of the central dogma, definitely the first half of the central dogma is harder for me than the second half of the central dogma. Okay, cool. I understand. Understanding is happening. 1, 2, and 3. Primary. This is the only known exception. Inaccessible. Accordingly, the cytoplasmic and nuclear. A picture of the measles 5' cap would be appreciated. I think it's just 3 P's in a row. Phosphate, phosphate, phosphate. Which would not be very interesting to draw, right? Just P, 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 I think. Although I don't know if they have a picture in this paper. And then in the eukaryote, it's attached to some other things in my illustration there. But I think I'd go with an analogy in the video. Okay, cool. Okay, so I understand. So that makes sense. A 5' triphosphate-ended RNA in the cytoplasm thus represents a well-suited feature for a danger signal, i.e. a molecular pattern that is absent from uninfected cells according to the concepts. Okay. Wakarimashita. I get it. That certainly makes sense to me. Okay. RIG1 is required for the recognition of measles virus infection and sensing the 5' triphosphate-ended RNA transcript. Okay. Since RIG1 has been associated to the sensing of infection by other viruses in this family, we tested RIG1 could also sense measles virus. Okay, perfect. Because we used an artificial system earlier, we have the bits and pieces. Now we want to know whether or not this actually happens in a real measles virus. Makes perfect sense. To ensure a homogeneous defect in RIG1 of the cell host, we took advantage of the Okay. Like I said before, it's not great to use kidney cells for a disease that doesn't infect kidneys. So what is this What is this cell line? 7.5. What the heck is this? This is God. What are you? This is from Homo sapiens. I mean, that's great, I guess, right? This is something that came from humans. But what cell type are you? Hello? Tumorigenic epithelial-like cells. A practical surrogate for primary hepatocytes. Epithelial-like cells. I mean, I guess that's closer to a lung. Not quite, but maybe I'm not familiar with how you would do this more faithfully. If anyone knows if there's a good lung cell culture cell line, maybe this is a practical limitation, but it's still not a lung cell type. Liver cancer, I think. Yeah, so like in terms of, is this better than a kidney cell? At least it's epithelium like. That might be a minor improvement, but the thing that they're using the cell line for is this fact that they do not have a functioning RIG1. They have a debilitated RIG1 with a point. Also, the choice they use the word debilitated, it's just not something that you would see today. With a point mutation in the first card domain, which no longer produces interferon after infection by VSV or whatever virus this is, unless complemented by exogenous wild-type RIG1. Okay, so this cell line that they are using, so they suspect that the thing that is detecting this measles leader sequence is this protein called RIG1, which has been known to be a pretty important immune detector, immune sentry. In this particular cell line, this protein is broken, and they work fine when you give them the protein from outside. So if you inject the protein, or if you provide them with the RNA to make the correct protein, they function normally, but they are by default a little broken in that regard. Measles virus triggers the interferon response in human HA7 cells, but not in RIG1 defected HA7.5 cells. Okay, so that's kind of interesting. Although the accumulation of N-mRNA reached a similar levels in both cells after 24 hours. Okay, so measles virus appears to work just as well in between the HA cells that have the working detector and host cells that don't have the working detector, that's, I suppose, that's not unusual. However, the interferon response is intact when you have a functioning rig 1, and when you have a defective rig 1, then the interferon response is not intact. That makes total sense to me. That, I think, is good and expected. Accordingly, 7.5 fails to respond in the in vitro T7 transcribed leader RNA, so using the artificial system before. However, interferon beta is activated by the double-stranded RNA paralog poly-IC, showing that these pathways are totally functional. Okay, that also makes sense to me, right? This cell line is broken very specifically, and it can respond to other sources of inflammation, but in terms of just the measles thing, it needs rig 1 to do that. So rig 1 is breaking specifically the detection of measles in this set of experiments. The inefficiency of the MDA5 pathways to respond to measles infection is likely because the inhibitory properties of this virus family, the V protein or 5 protein. Moreover, co-transfection of rig 1 with leader has strong synergistic effects on the induction of interferon beta. So when you don't even include measles in the equation, you just add in the leader RNA plus rig 1 into a cell, they freak out. So that makes sense to me. I don't really know if I like that experiment. I don't really know if it's necessary, but certainly that does make sense. In contrast, the overexpression of rig 1 had very little or no enhancing effect on the INF beta production by poly-IC. So yeah, so poly-IC, it's a different pathway. So rig 1 is specific, it's acting specifically for the measles virus. 20.3 words per minute. We're getting back into it. The ability of rig 1 to bind to the leader RNA was then tested in vitro. 10 times more phosphate labeled leader were pulled down by rig 1. Oh, yay, we have a pull down. This is a type of experiment that I have talked about a lot before, and so we can talk about it again. So 4A. Pull down. Lovely, lovely, lovely. So actually, let's continue reading. Let's not get too ahead of ourselves. Let's read the context of the experiment and then get into it. So for those of you who don't know, a pull down experiment is like a fishing experiment. You have the lure, you have the bait, and then you have the fish. And I'll get into what those are when we get to that section. If this were a journal club, I'd pull up those slides, but just reading today. Okay, so they are trying to see whether or not rig 1 binds directly to the leader RNA. And to do that, they are going to do this technique called a pull down. And so in order to do that, they're going to take the leader RNA, mix it with the rig 1 protein, and then try to detect how much of that leader RNA is caught. Like if the leader RNA is the fish, then the lure is the rig 1 protein. And so in order to detect the leader RNA, they radioactively labeled the leader peptide by putting a radioactive isotope of phosphate on it, so it would be easy to detect. That's pretty clever. I like that. Usually you don't really see radio labeling too often anymore. To a mock co-immunoprecipitate, figure 4A. Moreover, binding is independent from the ATPase activity of the helicase. So you don't need the enzymatic activity of rig 1. So this is a very pure interaction. This interaction is mediated physically. It's touching each other. Whether or not there's a third player involved, we don't really know, but this does not appear to require the chemical action of rig 1. Rig 1 pull down of radioactively labeled phosphate-driven T7 mRNA is indeed poorly affected by the presence of non-hydrolyzable ATP gammas and an ATP defective rig 1 mutant. Oh, poorly affected. Okay, again, this is such a writing thing. Poorly affected, so not affected. So not affected. Okay, so I was like, isn't this the opposite of what they just said? Okay, yeah, so it doesn't matter if you shut down the enzyme. If you stop rig1, the enzyme from working, it can still bind into some degree with the leader RNA. ATPase defective rig1 mutant binds to the, okay, yeah, you break the part that does the enzyme. This is all saying the same thing. 3 different experiments, all showing the same thing, which is that rig1's enzymatic activity does not change its affinity for the leader RNA. Radio-labeled microRNA purified from VSV transcription are also specifically pulled down by rig1. So this is something that it shares with its cousin, VSV. However, the mono or triphosphate status of the 5' end of RNA does not strongly influence the RNA binding ability of rig1. So that's kind of surprising. So we've been going on about this triphosphate being the signal that the cell responds to, but apparently it's a little flexible. We call it promiscuous. So there's a bit of promiscuity with this enzyme. It doesn't seem to care if it's strictly triphosphates or 3 phosphates or 1 phosphates. In agreement with the reported ability of double-stranded RNA to pull down rig1. Moreover, the rig1 affinity for 5' triphosphate and monophosphate is similar, grossly judged on the basis of saline elution step gradient. Okay, so this is... they're admitting that they didn't really do this rigorously. They just saw how much of it washed out with the enzyme, which I don't think you can do, because we'll see the data, because I doubt that you can control for the fact that the relative abundance of triphosphate and monophosphate leader RNA, there just can't be like that level of control. But we'll see how they actually did it. Maybe it's not as bad as I'm judging it to be right now. Thus, the 5' triphosphate appears to be dispensable for RNA binding to rig1, but it is involved in rig1 activation. Okay. So let's go down to that figure 4. So the leader RNA doesn't bind to the active site, or it is more correct to say that you do not need a functioning active site to bind rig1. Witty I am, your statement is a conclusion, and this is kind of what I like to model in terms of like, what is skepticism? Skepticism is saying, just calling it as you see it. And so your statement of binding to the active site is not something that can be ruled out quite yet. I think you're right. I think that is the conclusion. But we don't have fully the data, right? It could still bind to the active site, but the active site might be allowed to be flexible. So there are 2 models for the way that proteins bind. Both of them happen. They're not mutually exclusive. There is the lock and key, which is the first model that everybody learns, right? An enzyme binds with its partner in a lock and key format where the structures are very tight. I gotta go now, unfortunately. I have exams to grade. Good luck with grading, and thank you for attending and helping me patch up my weak understanding of some of this virology stuff. Yeah, so I want to take a little time to kind of emphasize why this is skepticism. Lock and key states that there are certain enzymes that are very rigid, that don't really move all that much, and they work by fitting a lock into a key. And then we have the induced fit model, which is some enzymes work like Play-Doh, where they are somewhat floppy, and then when they come together with their binding partner, they will kind of absorb and change around their binding partner. So what this is saying is that maybe there is some structural flexibility in the active site that can still bind the leader sequence, right? So that is something that is possible, probable, or not probable. It is something that is possible, but not probable. So in terms of if I had to bet money, I would say that that was the case, but yeah, just maintaining skepticism is I don't want to make that conclusion. I'm just going to call it as I see it, and what I'm seeing is that modifications to the active site don't bother the binding interaction, right? They don't interfere, whether it's because the leader sequence is binding somewhere else, or whether or not it's because the active site doesn't have to be that structured. That has yet to be determined. Yeah, thank you for covering this topic. Thank you for attending the stream, Rai. So for those of you who are unfamiliar, Rai is another VTuber who streams on Twitch. Rai, if you could plug yourself, that would be great. I did end up plugging your channel on a different stream, and I don't know if links can be posted. Let me quickly just Google your channel actually, so I can give you a shout out. But Rai is a real neuroimmunologist. I've had lovely conversations with Rai about the field. So if you want to hear Rai talk about neuroimmunology from a different perspective, by the way, we actually, even though in name we study the same thing, the actual topic we actually found through discussion quite different, honestly. Very interesting other side of neuroimmunology. I guess it's kind of fun that you found your way into this particular stream, Ryan, because measles has been known to be neurotropic, meaning that you can get infection of neurons by the measles virus. In fact, that is one of the worst complications to have from a measles infection, is that measles can make their way into the brain, and that often results in terrible disease, right? And we're talking about lag times, right? After the measles infection is over, you could die from it 15 years later. So it's pretty serious. Go get your measles titers checked if you're not sure if you've gotten a vaccine. If you got a vaccine for the measles, you likely have lifelong immunity. But if you're worried or a little paranoid, it doesn't hurt to check. Measles, super bad disease, but we kicked its ass so long ago. It'd be a real shame if it were to come back in a meaningful way. I tend to focus on sickness behavior. Yeah, my stuff overlapped a little bit with sickness behavior, but sickness behavior is something that I am always a little afraid to talk about on stream, because the part of sickness behavior that I studied is controversial. I'll put it that way. I overlapped with a controversial aspect of sickness behavior as a study, right? Because sickness behavior can be malaligned, I'll say. So I find that we have to have a lot of responsibility with regards to how we communicate that kind of field. Well, that's terrifying. It is. Measles is no joke, but it's the perfect topic for a Find the Neutral video, because it is horrible, it is horrifying, it is so bad. But I think I can joke about it because it's so easy to beat. Oh my god, you just get a vaccine. And you get lifelong protection. It's one dose. It's the easiest thing in the world. So while you can get it, you should definitely get it while you can get it, right? That's why I feel like it's maybe okay to be more jokey in a measles video than it is to be in an influenza video, because not a lot of people die from measles now as they used to. It is omega preventable. So that's my measles soapbox. Measles are one of my nightmare topics as someone with a primary immune deficiency and limited understanding of viruses. Maybe I spoke too soon. Maybe I should be a little more careful with how I cover measles. You bring up a good point. Oftentimes, immunodeficiency kind of escapes these rules that I have for myself with regards to how jokey can I be about any particular disease. The answer is you shouldn't be too jokey about any disease, but I feel like it's a little more okay to be a little more lighthearted when it comes to measles, because it is more easily dealt with. But Rai, you bring up an excellent point, which is something that I have to consider while making the script, is that just because most people can get a measles vaccine and be chill, we do have to be cognizant of the fact that there are immunodeficient populations that cannot take those vaccines. And actually, like, thank you for bringing that to my attention so that I don't cancel myself on this video. That is so much needed perspective, honestly. So thank you so much. It is really, really bad, and we need enough of us to take that vaccine for it to not be bad. If you've gotten other immune-erasing viruses, can't hurt to get a titer check to see if you benefit from a booster. Exactly, because measles is one of these viruses that can just eliminate your immune system, right? I actually, I'll say it now because I have very little ability to delay gratification, but measles, I think the most poignant metaphor that I will make in the entire measles story is the fact that measles is like, actually, this is a good testing ground, because you can tell me if this is too dark or if this would be inappropriate for the Fighting the Neutrophil video, because this is genuinely kind of triggering to some people, but it is that horrible. So I'll preface, this is my trigger warning or whatever, but like, this is something that I've been stewing on, is that like a measles infection to me is like escaping a very toxic and abusive ex. Right? Measles, when you leave measles, you actually leave measles for good, right? Unless you get one of these horrible neurotropic variants, or your body chemistry just happens to enable neurotropism, in which case you could have secondary brain infection many years later. But for the most part, when you are done with measles, you have lifelong immunity. Your immune system is like omega trained on killing most variants of measles. Measles is not something that we typically think of being immunoevasive in the way that the flu is, right? The flu is always mutating. Measles, not really that good of a mutator, turns out. But the reason why I compare measles to an abusive ex is because once measles leaves you, your immune system is changed forever. In fact, the only thing that your immune system can think about from then on is measles. The populations of T and B cells that you have built up your entire life, your whole quote-unquote immune personality that you have accumulated for your entire lifetime, has been replaced by this all-consuming infection. So you can actually plot the diversity of T and B cells that you have. And after measles infection, it's like mostly measles specific cells, which I think is really dark. And to me, that's like the emotional truth of, because I'm very emotionally sincere with this kind of body of work, right? Like I resonate a lot. That's why biology is so important to me. It wouldn't be my emotional truth if I didn't find those kinds of angles, right? Because I have almost a spiritual connection to this study. But that's also really dark, and I don't know if that would be appropriate to put out in a full video. And I kind of want to hear from my chat whether or not you think that it would be too much in a goofy video about the measles. It's not goofy, but like, I'm a cartoon fluffball. Whether or not you think that that is an analogy worth making, or whether or not I should just keep it for stream, because you guys get like the more unfiltered kind of off the dome, more artsy kind of interpretations from me, I think, whereas the video is a lot more curated. It's a lot more thinking about the craft of what I should and should not say. So what do y'all think about that? Too dark or what? Striking, maybe? I don't know. I'm gonna let chat stew on that for a second while I go up and get my cup of water, because I foolishly started stream without the cup of water in hand. So I will be right back. Need water. It does feel a bit dark, but I do think it's a very direct way to show the effect of measles on the body. I think, like, what I have to contend with is what is the utility of of this analogy, right? And I need to weigh that analogy with I need a way to weigh the pros and cons of dropping something that dark, right? Because it would be really effective, I think, for people who are vaccine hesitant, maybe, to hear if they were vaccine hesitant and open to change, right? Because some people are arguing from an ideological point of view, in which case there's no convincing them to take a vaccine. But there are people who are genuinely weirded out by the idea of putting a needle in their arm. And so drawing an analogy with the measles vaccine to something like an all-consuming abusive ex-partner gives it a kind of emotional weight. I was struck by this emotional weight because when I was reading review papers, there's this haunting data graph that represents the diversity of T and B cells with color, right? You know, and the more of that color there is, the more diverse those populations are. And then after measles infection, that graph changes color. It's like it goes from like blue and yellow, and then it gets replaced by this dark red to show that the color of your immune system has been monopolized. And a part of me thinks that because that is put in more of an emotional context, that should give measles an appropriate amount of scariness to it. The question, I suppose, is I think my viewers already get it, right? They don't need to be fear-mongered to. Although you'd be surprised, there are a decent number of YouTube comments that have gotten on my videos who are still anti-vaxxers, you know, COVID skeptics, etc., etc. So I know that's not the majority of the people who watch me, but they do watch, and I structure my channel in a way to be open-minded, right? In terms of I deliberately rewrite parts of my scripts that talk about vaccines to not be confrontational with people who are vaccine hesitant, right? Because they need to hear this stuff probably more than any of you do, honestly. And to be aggressive in that way of like, just take your vaccines is like not the angle that I want to take for the measles video. But at the same time, like the majority of people are here to listen to me talk about the coolness of the pathology, right? Or the study of it, right? The academic nature of my videos. And so implementing this emotional angle, I think, is like contains risk too, because it is an unpleasant kind of thing to bring up, and you know, when you're just watching a cotton fluff ball. Really interesting. I didn't know about the immune cells. Sets the direction of immune system survival instincts. Yeah, I'm used to sitting with heavy stuff, so I'd be okay with it. But I think other people may be sensitive if they happen to be triggered by trauma from lived experience. I agreed about live chat suitable. Yeah, that's kind of the vibe that I'm getting. Maybe I would recreate the data figure, and in doing so, I can allow people to come to their own conclusions. I think that might be the most sensitive way to do it, because the abusive X is my analogy. It is what I viscerally felt. Like I viscerally felt like my personality and my being has been overwritten by this infection, by this relationship. But if I were to just show that to people, just that data, that data figure, people might be able to come to whatever conclusion they are most comfortable with concluding themselves, or, you know, they can take it upon them to interpret the data whichever way they want to. Maybe that is the more sensitive option. I should probably write that down on my notes page just so I don't forget. Don't do the ex-abuser analogy. Just show the graphic. I think that is smart, and thank you for offering me your feedback. I saw here being off topic. For some reason, the Greek word for measles, ilara, which is feminine form of happy, cheerful. It's actually related to English hilarious. Apparently, it's a euphemistic name for the disease. That's kind of like gallows humor, I assume, because it's not great. But yeah, okay, so let's continue on. Tanking my words per minute. Let's see. Where were we? Oh yeah, talking about the pull down. Okay, so in this experiment, they're using the RIG1 protein, which comes from the host cell, to fish out the measles RNA. So when you have a, what is this? Anti-flag cytoplasmic extracts overexpressing incubated or transcripts by polymerase, Western blotting on the beads. Okay, yeah, so anti-flag M2 antibody used to assess the precipitation of RIG1 on the beads. Okay, so this is detecting the leader RNA, and then we have our different control groups here, or different experimental groups. So we are using the eukaryotic sentry protein, trying to fish out the transcript. So when you don't have infection, you don't get any of the leader sequence. So you know, this is essentially, this column is essentially not using a fishing rod. We use a fishing rod and we catch the measles RNA. Great. ATP gamma minus. So when you don't include, I believe this is hydrolyzable ATP. So this is non-hydrolyzable ATP. Essentially, this is, you're using the fishing rod, but the fishing rod is a little different. It doesn't come with all of its bells and whistles, and it still works to fish out that measles RNA. Oh my god, and then my pet peeve, dude. Control. Okay, what is control, dude? You better, I'm gonna be so mad if you do not define what you are calling control, because you already have mock. So what the hell is control? Cool, dude. Oh wait. Okay, red FP proteins as control. Okay. So just call it red DFP. Okay, whatever. Okay, so RIG1. Okay, so this is not even using a fishing rod, right? So this is not using a fishing rod at all. This is using the fishing rod that you think you should use. This is using a slightly different fishing rod, and then this is using a fishing rod but for different bait. I think that is the best way to describe that. Okay, so they do. Okay, so why is this a pet peeve of mine? Mock is also a control. So mock is a control that you do where you do everything the same except for the one experimental thing that is different. So for something like infection, a mock control is you do everything the same, and then for one group you put on your virus, and then the other group you don't put on the virus. But a mock infection, you put on the virus plus what the virus is dissolved in. That's your experimental group. And then a mock infection is you only put the thing that the virus is dissolved in, right? And that's already a control. So please, the future scientists of chat, stop putting control in your graphs. Just write what the control is. Here, instead of control, you could have just written red DFP, which is just a different fishing rod. And that would have been fine. It's like one letter apart, right? Like it's not even, you don't even save that many letters by calling it control, right? What, 5 letters here, 4 letters here? Like, come on. And I think earlier on in the figures, they were better at calling them control, like not calling them controls, right? Because here you have every group defined, even though poly-IC is also a control, it's a positive control, right? And then in this figure, yeah, did they call control here? No. They called this PBS, although this P is lowercase, which if you were there for the stream, that's really annoying. But the idea here is if you're going to make a figure, don't call it a control. There are all sorts of different controls, and it's as a unit of communication, just calling it a control in your graph doesn't actually help your audience understand it at all. Just label the dang thing. Thank you for attending my TED Talk. There may be a different analogy you come up with, though, that works as well or better. I had to swap one recently for my dissertation, and it took some time to come into mind, but it was better replaced. It might take me a while to fall out of love with that comparison, just because I do find it so emotionally resonant. Heading out, have a good stream. Thank you so much, Mylan. Have fun doing whatever it is that you are going to do next. When you have control 1 and control 2, don't. No member of my audience will ever write control 1 and control 2, man. Do not, man. That is heresy. But yeah, so this basically shows you that rig 1 is a fishing rod for the measles RNA, and you don't need the fishing rod to have all of its bells and whistles for it to completely work. And I'm pretty sure that's what the rest of these are saying as well, so we can move on from that figure. Where were we? Pull down, blah, 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 10 times, no significance bars or anything absolutely crazy. Papers from 2007 really do be built different. Talking about rig 1 mutants, this is, again, if you change the fishing rod just a little bit, it still works. And it doesn't matter if the cap is mono or triphosphate, so interesting. Modeling of the rig 1 helicase domain and prediction of Q99 as an RNA contacting residue. Okay, so we don't know what part of these two molecules are interacting with one another, right? We do know that the fishing rod works. We do know that the fishing rod doesn't depend on enzymatic activity, but where and how do these molecules interact? That's what this is referring to. In order to obtain mechanistic insights into the rig 1 interaction with RNA, we searched the Protein Data Bank, PDB, for possible structural homologs. This led to the identification. So structural homologs basically means we looked at the Protein Data Bank to look for proteins that are similar. For possible structural homologs of the RNA or of the protein? I assume it's the Protein Data Bank. Okay, so they're looking for proteins that are similar to rig 1 to see if they can learn from related proteins. So they found a related protein called HEF, a helicase from Pyrococcus. I assume that's a bacteria. Furosis. That's kind of a fun. That's kind of a fun name. That shares 43% similarity and 25% identity with the helicase domain of rig 1. So they found a match ? 43% is kind of low. But okay, fine. We used it as a template to build a homology-derived 3D model of the helicase domain of rig 1 5b. Okay, so this is in the days before AlphaFold, where you could just feed an amino acid sequence into AI and then have it develop a model for you. You actually had to be a lot more clever. So they took their raw information and they played with some models in order to get some structural insight. And so we will read what kind of structural insight they got so that we can put that figure into context. In order to identify the rig 1 residues possibly involved in the RNA binding, we performed a structural alignment among the rig 1 model and the crystal structure of prick A DNA helicase from Bacillus stereothermophilus. It's so weird that that both of these homologous proteins, so when they were looking for rig 1 relatives, I can't believe they had to look all the way into bacteria. That's kind of surprising to me. Rep from E. coli. Hey, look, shout out E. coli, Hep C virus, which all share an identity of 42% having crystallized with a DNA oligonucleotide. So this is essentially trying to take a shortcut, right? Like, they don't want to do the crystallization. That's totally fine. There is a reason why Rosalind Franklin is often said to have deserved a Nobel Prize alongside Watson and Crick, because crystallization is horrible. You know, you have to have a very dedicated, very clever, very smart, very careful scientist in X-ray crystallographers, right? And so this paper is saying, hey, we don't have a crystal structure for rig 1 plus RNA, nor do we want to nor do we want to be the ones that actually do that. Which, by the way, totally reasonable. That's not a slight to them. If you don't want to do a crystal, nobody's making you do a crystal. So they're doing the next thing, which is doing computer things to try and figure out some insight which you can then test. So this is not making a structure. This is, we are going to make a hypothesis based on information that we currently know. And this is, again, the days before AlphaFold, so you can't just predict a model, right? I wish I understood any of this, but I don't have enough basic knowledge for it. That's okay, Diamond. This is purely me interpreting professional level literature, right? If you come in and you don't understand, that's extremely normal, because this is stuff that working scientists read, and of course it's going to be opaque. You should return for the measles video that I'm hoping to get done in September or August, somewhere in that time frame. I'm hoping to make a measles video. Right now, I am interpreting this information for the first time, so it is very, very high level, but I will translate it to be more accessible when that video comes out. Structural bio mentioned. Structural biologists scare me. Is it like simulations? Kind of. I mean, as good of a simulation as technology in 2007 can do. Okay. Although NS3 has a more divergent primary sequence, it has an overall fold very similar to rep and PCRA. So, okay, even though the sequence similarity is not super good, it looks like RIG1 has important parts of its structure that are similar to these other helicases found in these other organisms. Among the nucleic acid binding motifs of helicases, namely 1A, 3, 4, and 5, the motif 1A is devoted to oligonucleotide binding, while the other motifs have additional and not yet well-defined functions, which is kind of funny because I'm sure that the rest of these motifs in RIG1 have been figured out. Okay, but motif 1A, this is some part of helicases. That's the part that makes contact with RNA. So this is the part that we're probably going to be interested from here on out. As these motifs are divergent in their sequence, the oligonucleotide binding residues are not conserved. So this is speaking from an evolutionary point of view, right? Like, we know that this part of the helicase is likely very variable, that evolution did not keep this the same across organisms. So that's why they're saying that this sequence is different across different proteins. Moreover, even when the motifs are strictly conserved, as in the case of motif 1A, FTN, KAA, and PCRA and rep, the residues involved in DNA binding are not the same. So even though these two helicases, one from, Bacillus and the other from E. coli, even though their motifs are the same, in the context of the whole protein, the actual individual amino acids here that bind DNA themselves are not the same. So this is basically all building up to the fact that this family of proteins called helicases are very, very variable in how they are structured. In addition, despite the overall very similar fold shared by these helicases, the bound oligonucleotide molecules are not superimposable, data not shown. Again, the data not shown. Okay, I guess back then they were printing a lot more of these journals, so word count was probably a lot bigger of a deal. Although these latter points highlight the divergence of helicases in their mode of binding to nucleic acid. Yeah, in terms of like whether or not people understand it, I don't expect anybody without 4 years of biology to understand these things. But it's just fun to have you all around, and I think it's fun to share with you the process. So if you're here to just vibe and be in the same room as someone trying to fight for their life and trying to figure out what this paper is saying, I think that's also, there's value in that as well. There's also educational value in that as well, because I'm also, I think, modeling a lot of what I assume to be good practice with regards to reading a paper, right? Skepticism, but I'm not cynical, right? Practicing skepticism, but not practicing cynicism, etc., etc. And that's all you get. You also get like sneak peeks in the measles video. So now when you watch the measles video, you can go, hey, it's that what is it? That meme. Hey, I know that guy or whatever. Hey, I recognize that meme or whatever. Now when you watch this stream, you can recognize the parts of this paper that I will put into that proper context, because let me be real, most of this does not make it to the video. I did write down some things that are worth elaborating on, but this is the research process, right? Like, you read a whole dang paper and you pull 3 sentences out of it, the 3 of the coolest sentences. Why are helicases not well conserved? Are they less specific than other enzymes or something? That's an excellent question, Mr. Winner. I don't know, because you would assume that anything having to do with touching DNA would probably be pretty well conserved, but I don't know. It is a really good question. I would assume that helicases are quite well conserved, so my assumption was proven wrong. And oftentimes, the best parts of learning biology are when your intuition is incorrect. Okay, in order to identify the RIG1 residues possibly involved in the interaction with RNA, we analyze the residues involved in the interaction with the oligonucleotide in 3 helicases and their structural components in RIG1. Okay, so in order to determine what they should be looking at in RIG1, they are taking a look at other similar helicases, and because they have crystal structures, they're using those crystal structures to generate hypotheses about RIG1, saving them time, because you could do something called an alanine screen. I made a short on this when I was doing a tier list of lab techniques. An alanine screen is when you just take a sequence of DNA that codes for a protein and you just mutate each base one by one to an A to break the protein in every amino acid one by one. And yes, doing an alanine screen would certainly work. However, each alanine mutation you do is a metric butt ton of work, right? I said in that short that alanine screens are built upon the corpses of graduate students, right, just piled up on high, because making one mutation per amino acid in a protein, like, look at how complex this thing is, right? Imagine having to mutate every single individual amino acid down this entire spiral. Each one of those mutations is going to require a separate bacterial culture, which is going to take at least a week to do, let alone have it succeed. It's just a nightmare. So they are using other structures that look like RIG1, enough like RIG1, and they know where DNA, so like these are the other helicases, right? You can actually see it. These are not RIG1, but these are the RIG1 homologs. So you can kind of see that the predicted structure, right? That's what this is, right? Blue is predicted, right? Surely. Cyan. Yeah, the RIG1 structure is in blue. And then you can see that there's generally pretty good overlap with the other helicases. And then down here, you can see the DNA binding site, right? So this right here, this is where this structure binds DNA. This is where this structure binds DNA. This is where this structure binds DNA. It looks like the part of these helicases that touch the DNA are in these unstructured linker regions, right? These alpha helices, these are structured, and then this little thin noodle that's unstructured. And it seems like across these different things that look like RIG1, that there are amino acids within this spaghetti part that are important for DNA binding, although it is different for each structure. Yeah. So this is saving them a lot of work, right? Instead of having to mutate every single amino acid one by one, they're only probably going to mutate amino acids just in this one turn that they suspect is important for DNA binding. So smart. Very smart idea. I lost my place. Okay, we analyze the residues. Okay, in rep, 2 residues interact with DNA, namely these amino acids, sure, 56 and 58. In RIG1, the only residues close to the DNA molecule crystallize are GLN. Which one is GLN again? Gluta. No, I'm forgetting. I'm blanking. Well, pro is proline. That one's easy. This is gluta. Not gluta. Is it glutamate? Glutamate or the other one that isn't glutamate? What is it? DNA binding, not RNA. It is a bit of an unusual thing to look towards, but I think that what do I want to say? RNA and DNA are similar enough where I think this is an okay strategy. Because RIG1, shoot. No, you bring up a good point. Glutamine, thank you. Not glutamate. It's glutamine. Yeah, glutamine. Just blanking on that. But witty I am, you bring up a really good point. We're talking about RNA because it needs to detect measles RNA. So why are we looking at DNA? Is it because DNA and RNA are similar enough? In order to obtain mechanistic insights into the RIG1 interaction with RNA, we searched the PDB for structures. We got these other helicases. Helicase domain of RIG1. Because helicase is DNA, right? You don't need to unwind RNA, but you do need to unwind DNA. Okay, so maybe that answer is here. While the crystal structure of HCV NS3 contains an RNA molecule. So I think it is similar enough to other helicases that bind to RNA. So it is kind of a shot in the dark here. Because, yeah, I think it's because DNA and RNA are structurally very similar. So they're hoping that that's enough. But that is an excellent observation to make. It's not exactly one-to-one. Gotta go. We'll do the rest of the words per minute from VOD. Have a good rest of your stream. Thank you so much, Astral. I will give you a timestamp for when I end to make your life a little easier. But thank you, as always, for doing the spreadsheet. It's super entertaining. For those of you who don't know, Astral has been keeping the V-Theorem words per minute tracker alive. And I'm trying to defend my title as most words per minute V-Theorem member. Words of the paper, not words overall. Okay. Like I said, when you get a bunch of academics in a room, there's no way that we aren't going to be a little competitive in some kind of way. Okay, so in those other proteins that are similar to the fishing rod that we're using here, we have two of these candidates, this glutamine 299 and proline 301. These latter residues are close enough to establish contacts with the DNA molecule of the PCRA DNA complex. Okay, so this checks out when we compare with their cousins. Although it's slightly different because instead of the glutamine or proline, it's phenylalanine. Okay, cool. I mean, I'm happy with this, right? They're narrowing down the exact amino acid that they should be mutating to try to see if that abolishes the RNA binding. Within RIG1, there are three residues that could interact with the RNA molecule: tyrosine 303, isoleucine 300, and glutamine 299. Tyrosine 303 could establish an H bond via its OH group to the alpha phosphate of this RNA, and isoleucine 300 can interact either by stacking the ribose or through its backbone carbonyl or amide atoms. So that entire thing is for the organic chemist to figure out. The side chain amide group of glutamine can act simultaneously as a hydrogen bond donor and acceptor. Glutamine 299 is the polar residue closest to RNA and could more specifically interact with the nucleic acids. So they are hypothesizing that it's probably glutamine because glutamine has chemical properties that make it more favorable to interact with RNA, I believe. That's so fun. It really is. I am the reigning champion. Not that it really means anything. I mean, at the end of the day, we're all streamers who make content, and what matters is whether or not the content is fun and entertaining. But you get a number for us to compete on, and that's all we're going to be doing, to some degree, right? To some degree. In conclusion, the structural comparison led us to predict glutamine 299 is the best located residue to specifically interact with RNA. Accordingly, we designed and constructed a RIG1 glutamine to alanine mutant, right? They're doing that alanine scanning mutation, and found that the mutated protein is unable to bind significantly to bioattenuated leader RNA, figure 5G. And then we're in the discussion section. Wow, this paper just kind of ends there, huh? I mean, this is kind of a major experiment, and they say a total of one sentence about the results. That's actually kind of crazy. Okay. 5G. So, unbound. So this is what? A pull down of some kind. Binding assays were described in figure 4 legend. Cool. So we'll go here. So how is this done? Okay, so this is co-amino precipitation. So they're using the fishing rod technique, and they are fishing for what? What are they fishing for? They are they're detecting RNA, and they are using, what is unbound? Eluded, this is the thing that we really care about. Oh wait, okay, so this is likely just checking to see if the fishing rod is there. So it's there, it's the black band here, there's a fishing rod of the mutant rig 127DA and then Q299A. And then you do the pull down. So when you have the normal fishing rod, it's chill. You get the measles RNA binding, which is represented by this black band. When you break the fishing rod in a place that is not the predicted binding site, you still get successful fishing, which is great. Q299A, this is the amino acid that you suspect is binding RNA. When you remove that, you no longer are able to fish out the measles RNA. So it looks like we identified exactly which amino acid, down to the exact amino acid on the RIG1 virus infection detector that is responsible for detecting measles, which is super, super cool. And then DSR. Okay, what is this? DS red and mock. Is this, what is this? What is these? What are these? Dude, what is this? What is this? They don't mention it. And as you can see, for figure 5G, they literally have one sentence. This is the sentence. It doesn't bind the leader RNA, but then they don't freaking explain what this control is. Is this double-stranded RNA generally? Like, what is the sequence of this double-stranded RNA? It wasn't, you know, I can't find this abbreviation anywhere on the paper. And then, what is this? This 0 with a cross through it. Phi looks like. What is that? And why is it weaker? God. This makes me so mad, dude. That's okay. 2007 was a different time. PLOS was a different journal back then. I get it. But still, man, like, I can't even easily copy and paste wherever this is supposed to be. This is the Greek letter phi, right? Am I silly? How would you say it's that one in more than one sentence? I would define what the controls are and tell the complete story, not just say the main conclusion, but also explain some of those controls. Phi Greek letter. Yeah, it is phi. It is the phi. So let me control F to see if we can figure out what this control is. Nope. What about lowercase phi? You gotta tell me what that is? What about lowercase phi? Nope. What about the other lowercase phi, huh? How about that? Nope. Okay, so total mystery control here. I have no idea what that is. Anybody in chat want to offer some insight onto that? It's the empty set. Shouldn't you know your name? I've said this before, but if you're a human, do you know everything about being human? That's my go-to. That's my go-to excuse. But yeah, I have zero idea what these two mystery controls are. They do not appear to be elaborated in the body of the text. Maybe they talk about it in the discussion, but maybe it's here? They said refer to figure 4, right? Nope. No, not in there. I mean, maybe it's in the methods, but I controlled F, so why would it be in the methods? Transfections, purification, qPCR, monophosphate leader, modeling. Okay, immunoprecipitation. Unlabeled, 30 minutes, scintillation counters. Immunoprecipitation, comparable sizes, radio-labeled. Yeah, no, it's not there either. No. We're going pull down, but bioattenuated RNA. All the buffers, mixture was stirred, stripped evident, blocking buffers. This is just all regular pull down stuff. Wash buffer, elution. Yeah, nothing about the individual groups. So this is just an orphan. This is just orphan data. It's just orphan data. Cool, dude. Google says that the phi stands for diameter. I don't know, man. I don't know. Wow, someone reading a paper? Too much for my brain. Welcome to the stream. This is what I do somewhat regularly for, you know, the research I do for my channel, and I figured I might as well make content out of it by sharing with all of you. What does pull down mean? So a pull down is a colloquial term for co-immunoprecipitation, which is the act of using one protein as a fishing rod for another binding partner. So in this case, what we are pulling down, we are using a fishing rod. The fishing rod is the RIG1 protein, RIG1 being the immune surveillance protein, and we are trying to fish the measles RNA out of the cell to see if they bind together. So what this is saying is that when you have the normal wild-type functioning fishing rod, you are able to find the measles RNA when you have a measles infection. When you mutate your fishing rod in a place that you don't expect to be in the DNA binding domain, you still see that the fishing rod is working, right? Like, just because you tore off some aesthetic stickers on it or something does not make it a fishing rod. Then when you break the fishing rod in exactly the same place where you think that fishing rod binds to the bait, in this case, the measles virus RNA, you no longer get that RNA being pulled down, right? You no longer have a band here. And then I really wish I could explain what these are, but I super can't because it's not mentioned in the paper. So that's chill. Diameter, maybe. Yeah. All right, so what do I conclude before I read the discussion? And by the way, Mylan has a video about tier listing, APA, style guide stuff, which is really funny. And Mylan put discussion in A tier. I actually put discussion, I would say, in C tier because I often don't want to be poisoned by the author's opinion. So I often skip discussions. And maybe that's not the right thing to do, but I typically don't even read this section. But for you all, and to keep the spreadsheet fair, I will read the discussion and sort of see where... Okay, but before I do, before I am poisoned by the author's interpretation, let me share with you what I think. And if we happen to land on the same conclusions, that means that we agree in large, right? So I think that just from this cursory reading, you know, I didn't do a tremendously critical reading. If I did a critical reading, it would take me quite a lot of time to make sure that I did a good job. In a cursory reading for a video, I'm not going to be that critical, but upon first glance, here's what I conclude. The measles virus is very sneaky. A lot of what makes the measles virus does not upset the cell at all, right? The measles proteins don't appear to elicit an interferon response. A lot of the mRNA, the messenger RNA that becomes protein, does not elicit an immune response. But what does elicit an immune response is this non-coding leader RNA, is what they call it. This leader RNA contains a 5', meaning it's on the 5' side. Don't worry too much about it if you don't know what that means. But on one end of that RNA, it has a little hat. This hat is the triphosphate. It's 3 phosphate groups connected to one another. It has this unique hat, and it is this unique hat that the immune system uses to detect the presence of measles. You can, if you remove the hat, this doesn't work very well. If you change the RNA underneath the hat, it still works fine. So it looks like this hat is really important for the immune system. What's also known is that the protein that is detecting this leader RNA is RIG1, and RIG1 needs this particular amino acid at position 299 called glutamine. It needs that to be glutamine. If you change it to alanine, it doesn't work anymore. So we've actually gotten a couple of very interesting pieces of information from this. We now understand that in this line of kidney cells, what the initial signal flare is for the immune system. And again, I do have to mention repeatedly that this is in embryonic kidney tissue. This is not in its more typical niche of the respiratory tract, nor is it in the immune system, which may or may not matter, but it's important to keep that in mind. So we know what the signal is, we know what the signal isn't in kidney cells, and we know how this signal is connected to the sensory machinery of a typical mammalian cell. Now, of course, this comes with a ton of caveats, right? Didn't use a respiratory cell line, didn't use a human wait. Okay, no, it did, right? Hex cells as human embryonic kidney. I was gonna say, whatever you do with measles, it has to be in human cells because measles is a human-only disease. It does not have a wide species tropism. That also means, as a side effect, that every time you see a mouse study using measles, yeah, gotta take that with a huge, healthy grain of salt, right? And I will make that clear in my measles video. Human-only diseases are kind of hard to study because you don't really know what you can conclude from animal models. Not to say that science is useless. It's very useful, but you have to be like very, very careful with what you consider to be true for humans, which I think is interesting. So those are my conclusions based off of what I have read. I liked your videos a lot, so I'm here. But what's 102 kDa at least? I mean, as a unit. 102. This is kilo. Sorry, that's a great question. So this is kilodaltons. It is a measure of size. I don't remember what 1 kilodalton refers to, but this is basically how big the whatever you're fishing out actually is. Yeah. I think I got it a little confused. I think they used the RNA as a fishing rod and then fished out the RNA is the fishing rod, and then the fish is the RIG1 protein. I think because the RIG1 protein looks to be about 102 kilodaltons long, 102 kilodaltons big, I should say. So I think, everything I said about the pull down is true, but I think what we are visualizing here is not RNA, but rather protein. I think that only matters if you are an actual biologist going over my work. I've also seen this in linguistics where it stands for nothing. I can imagine because in math it's the empty set, right? Does this leader RNA originate from the virus or from other cells? If it comes from the virus, why would it make that? So this RNA is from the virus. Why would it make that is an excellent question. I do not have the answer to that quite yet. Why does measles make the leader RNA? I can say in the general sense, Mr. Winter, is that leader RNA sequences are useful for stabilizing RNA. So as I've said in multiple videos, RNA, I joke about this a lot, but like if you're a grad student, you're working with RNA, if an undergrad sneezed in the following room, all of your RNA would degrade, right? That's kind of my shorthand to be like, RNA does not want to exist. It breaks down very easily. It gets chewed up by all sorts of enzymes. Adding a cap to the RNA sequence gives it a little more wiggle room. So your normal healthy RNA also has sequences that bookend the RNA to prevent it from getting destroyed. A lot of your sequences contain what's called a poly-A tail. So you have your gene and then it's made into mRNA, and then the mRNA that is made has a tail that's just a sequence of A's. A. Those A's are there to buy the molecule time as it's being attacked by all sides from all sorts of different enzymes to make it to the ribosome to be made into protein. So if I had to guess why this leader RNA sequence gets made, it's likely, I think, to stabilize the real mRNA, right? Because the leader RNA is by definition not mRNA. It's a different type of RNA that does not get made into protein. mRNA is made into protein. Leader RNA is just there to stabilize the RNA. I think that's my hypothesis. Excellent question, though. I did write that down to address in the actual video. What about experimenting on something like lab-grown human cells or something? Why don't they do that for human-only infectious diseases? They do. However, cells in a dish don't have an immune system. That's why we still need mice, right? I mean, the mice aren't going to be perfect because their immune system doesn't look like ours, but if you, like in this paper, right, they used human-derived embryonic kidney tissue. And that means that those cells look a lot like our cells. But if you want to understand what a virus does, you need to look at it from the context of the organism, because ultimately it's not cells that get infected, it's humans that get infected. So I've been reading about measles, and this is not the first measles paper I've read, but it's kind of interesting that there are ways in which a measles infection happens in mice that just categorically do not happen in humans. And they have a different disease when you give them human measles. Like the symptoms are different. So even though mice are not perfect because they don't actually have that much relevance to humans, they still are an organism that can get infected, that can respond with an immune system. And when we take that knowledge that we gained from mice and we compare it to what we're seeing with humans, we can kind of see what we can keep and what we can throw out. So I'm very much an advocate for animal research for human health. It's unfortunate, but true. You just need something with immune system to fight it, to see how it reacts. Otherwise, you're going in with no information, and some information is better than nothing. Yeah. Am I silly, or are the two results aren't very connected? They determined the crucial amino acid in the helicase and showed that the 5' triphosphate is important for the interferon response. I just don't see the connection. We would be on the same page with that. I think that there is not really that solid of a connection, but it is tied together in the story of what is the mechanism by which the cell, not the immune system, but the cell, responds to measles infection. And again, this might not even happen in a real organism, right? Maybe the immune system signals to the cell immediately and cuts this off at the head, right? Like, there are so many ways in which when you move to an organism that all of this stuff just ends up not being true, but you still have to do it anyway, because you need to be working with some information rather than working with none information. So that's kind of my soapbox for continued experimentation with animal models. Now, it's kind of an interesting segue, because I do have to acknowledge this, that do I think AI can replace animal models and save a lot of animal cruelty happening in the process? I think AI can reduce the reliance on animal models. I think that there will be more wise uses for animals with the implementation of AI technology, but AI is only as good as the information you feed it, right? So assuming that we have not solved mice or solved humans in terms of knowing every little thing about them, the AI itself will also be imperfect. So to supplement AI as a research tool, you still need to test, but you can test fewer times because AI can make your hypotheses more worth testing. I think that is a genuine use case for AI here. Okay. That being said, let's read the discussion. Gotta take a drink of water first. Talking a lot. All this yapping. The bioinformatics analysis of the RIG-1 sequence, as well as the RNA binding experiments, confirm previous findings. Okay, so already this is oh, sorry, here it starts here. Our present findings strongly support 5' phosphate-ended single-stranded RNAs as being a danger signal capable of inducing cellular innate immunity. These single-stranded RNAs are recognized by the helicase RIG-1. Okay, yeah, that's a pretty good top-line summary. I think I agree, probably. Again, not that critical, but that is the feeling that I get here. Okay, so how does this work? The mechanism. The bioinformatics analysis of the RIG-1 sequence, as well as RNA binding experiments, confirm previous findings, indicating this molecule belongs to the DXD XD hRNA helicase family. RNA binding is mediated by the helicase domain, and we showed that it involved the glutamine 299 within the helicase 1A motif, while the ATPase site does not play a critical role. RIG-1 has been reported to bind to many different ssRNAs and dsRNAs in this work. I can't just worded weirdly. Using a physiological RNA target for RIG-1, the measles virus leader RNA in vitro RNA binding to RIG-1 was found not to be significantly enhanced by the presence of 5' phosphate end, in contrast with recent reports for Hornung et al. And Pilchmeyer et al., who carried out similar assays. Possible reasons for this discrepancy could be that the longer single-stranded ssRNA we use compared to those used by them could obscure the contribution of the 5' triphosphate end to the RNA binding. Pitchmere et al. Used a much longer RNA and a RIG1 GFP fusion protein instead of flag-tagged RIG1 protein. So their experimental systems differed. That could explain some discrepancies. So they're paying attention to whether or not this fits in the context of the other research that exists here. We could speculate that the presence of this GFP tag may lead to subtle conformational changes of the helicase domain, possibly by modifying its post-translational modification through changes in the cellular polyubiquination system. Since alanine substitution of... Okay, so this is a super common technique, right? They used RIG1 GFP. This is attaching a fluorescent molecule at the end of a protein that you want to study in order to be able to more easily track it and more easily purify it. This is a very common practice. And you always have to keep this in mind, that whenever you make a change to a protein, there is a chance that you screw up the protein's natural function. So here they are being good, and they're saying, you know, we used a slightly different system. Or they use a slightly different system. Their use of a GFP tag might have changed their protein a bit more, may have changed it in a different way than our protein. We used a different tag. Since alanine substitution predicted to bind to the RNA chain and not to the 5' phosphate end abolishes the right ability of RIG1 to bind to RNA, we propose that the 5' end of RNA only marginally contributes to RNA binding. While it is critical for RIG1 activation and exposure of CAR domains for binding to IPS1, indeed, isolated CAR domains from RIG1 act as a constitutive activator for the IFN response. So this is actually going back to what WIT-IM has caught on to. Like, this proves that WIT-IM, you have been paying attention, maybe even more attention than I have. Because there is this nuance of like, this whole first half of the story about the 5' triphosphate end doesn't really have to do with the RIG1 story at all. We know that in order to induce an interferon response, you need that 5' hat, but RIG1 doesn't need it. RIG1 needs a different thing to be able to bind to the leader sequence, so there's a piece of the puzzle missing, and I think WIT-IM's lack of a connection, what you are seeing there is true. There is a lack of a connection. There's a story beat that is missing, and that is, okay, if the 5' cap is required, what is the 5' cap doing? The 5' triphosphate end. What is that 5' triphosphate end doing if not mediating binding to RIG1? Because you're right, there is no connection there. This is two separate things. Like, you have the beginning of a story, the ending of a story, but you don't have the middle. So that is why it is a very understandable feeling to feel like there's something missing, because there totally is. Short uncapped measles. By the way, that's not a problem with the paper. I don't fault that for being the case. That's just what they found, right? And then they published. So I'm not like upset about that. Hi, Seon. Welcome, welcome. We are in the discussion section, finishing up this paper. As other mononegavirales, measles virus has a polymerase producing two types of 5' triphosphate RNAs, the 15,894 nucleotide long genomic and antigenomic RNAs, and the short leader RNA, which can be recognized by RIG1. Okay, so this is a bit of a pet peeve. Two types of triphosphate RNAs. Genomic 1, antigenomic 2, and the short leader 3. I get what they mean. Antigenomic and genomic, they're lumping in the same category, but it still threw me off to read. So short leader RNA, I assume this is still attached to the genomic and antigenomic RNA, but as I wrote in my notepad, I need to know how the measles virus RNA is structured, because this does not appear to be your typical one-of-the-mill virus with regards to how its genetic information is structured. So I'm definitely going to have to do a bit more reading on that. The 5' triphosphate moieties of the genome and antigenome are not recognized, as shown by the inability of UV-activated measles virus to induce an interferon response and in our data. Likely because they're shielded within the nucleocapsid. Ah, did I get that part wrong earlier? When you break the RNA... More during virus replication. Yeah, when you break the RNA, it still works because it's the 5' hat that's important. That went, wow, hold on. When you use the UV here, you are crushing the response. So what are they trying to say? Huh? That's not what I would conclude. I'm confused. Our data, you could point to it, dude. You could reference it. You could reference it like you do elsewhere. You could point to it because as shown by the inability of UV inactivated measles virus to induce an interferon response because they're shielded with the nucleocapsid. What? Are not recognized by what? Are not right? Huh? I'm recognized by Rig 1? I don't. If you couldn't tell, I do not like how this paper is written. I do not like how this paper is written. 2007, dude. Pain. Whatever. I'm just gonna move on to that. I don't quite understand what point they're trying to make there, but I'll revisit this paper at some point. Moreover, during virus replication, the simultaneous encapsulation by the N protein of nation 5' triphosphate genome antigenome is predicted to prevent them from being recognized by Rig1. So the fact that the RNAs are packed into this tight little protein package makes it so that Rig1 cannot detect measles. As a consequence, if, as recently described, the 5' triphosphate moieties of the naked genomes and antigenomes from rabies virus can be recognized from Rig1, it is unlikely to reflect the in vivo situation. Rather, we have proposed that the short 5' triphosphate leader is transcribed in the cytosol by all measles family members, but which replicate into the nucleus represents a danger signal that triggers the immunity and accounts for the massive and immediate interferon induction from infection. Sure, so the leader is sent off into the cytosol, and then that's what causes the danger, and that's what causes everything to crap out. Okay, so just for my own notes here. But isn't that always the case? Are there nuclear factors that detect RNA? I don't understand. Maybe it's me. Although 5' triphosphate ended leader RNA from the measles virus has the potential of being responsible for the activation of Rig1 dependent INF beta activation, it remains to be established whether the small RNAs as free RNA in significant amounts during. Yeah, that's kind of what I'm surprised by. Because this is saying that, like, you don't really get replication of the measles RNA before measles start causing the cell to freak out. So my initial skepticism very early on in the stream is, do you have enough starting material to actually cause something to happen? So this is saying it remains to be established whether this small RNA is produced as free RNA in significant amounts during virus transcription. So this is kind of saying we don't really know where this leader is coming from, if it's made, if it's like dropped off on the way into the nucleus, or if it's made in the cytosol. It's kind of a complicated idea that I'm going to have to think a lot more about. So far, measles virus leader RNAs have been found only as encapsulated read-through leader and gene RNAs, as they should be poorly prone to activate Rig1 based on the same arrangements as for the genome/antigenomes. My brain's not functioning enough to understand any of that, so I will continue. From the current model, mostly derived from studies done on Sendai virus and VSV, replication and transcription promoters of measles family viruses are always switched on, and their polymerases continuously enter at the 3' end of genomes and antigenomes to initiate RNA synthesis. In the absence of soluble N protein, the polymerase is poorly processive and produces small amounts of leader and larger amounts of leader N read-throughs, short RNAs. Oh boy, I'm going to have to read about all of that, which fall off the template because of the poor processivity of the polymerase. Okay, so this is kind of explaining why measles suck at making its own genome, because it needs to have a very specific setup in order for its own polymerase to do work. It looks like you need a helper protein, and when you don't have that helper protein, measles doesn't really work as well. Upon scanning forward and backward, the polymerase encounters the transcription promoter located downstream of the genomic promoter and starts the transcription of the first N gene. Okay. This is information I wish we would have in the background, honestly. Transcription becomes processive because nation mRNA is capped by the polymerase shortly after the initiation of RNA synthesis. Okay, so that's why... Okay, don't really see the connection there, but that's okay. Upon arriving at the next intergenetic junction, the polymerase stutters to add poly A, right? That's that poly A that we talked about. Stops and restarts to synthesize the next gene. Later in the infection, the availability of soluble N allows the specific encapsulation of the nascent leader sequence, which contains the encapsulation signal and switches the polymerase to a processive replication model. Indeed, most of the read-through leader N RNAs are also found encapsulated at later times in the infection. Boy, this discussion is dense, and it all feels like information that I could have used earlier. Because they're just saying stuff. This is not from the paper. This is citing previous work, and that would have been better suited for before I started to try to understand any of this. Although a similar RNA synthesis initiation can occur on the antigenome with the synthesis of a short trailer transcript, it requires the accumulation of antiviral genomes that begins 12 to 15 hours post-infection. According to this model, the ended leader sequences are continuously transcribed at a level similar to the transcription of the first N gene and as such remain the best candidate for the RIG-1 dependent simulation of, oh gosh. The lack of detection of such small non-encapsulated transcripts in the case of the measles virus may reflect their high instability or the rapid dismantling after their interaction with RIG-1. So that is a part that I think is interesting. The mystery of where the measles genetic material goes and is replicated. It's really bad at making copies of itself without protein help. Is this a feature of its own structural instability? Trying to extend meet word count requirements on an assignment at the end. I just think it's poorly written because this is interesting and good information. I just think it should have been in the introduction. Finally, from a coevolution point of view, our and other data suggest that mammalian cells have evolved so as to confine transcription of various RNA maturation processes within cellular organelles. As a result, this has enabled cells to develop new tools to detect any RNA synthesis occurring in the cytosol. Okay, that's interesting, right? This idea that processed eukaryotic host RNA looks different than processed viral RNA. They are each processed in different locations within the cell. Oh my god, I'm so tired. But we can do it. Last paragraph. Such as RNAs being 5' triphosphate in it are readily recognized by RIG-1. As a result, RNA viruses completing transcription in the cytosol have developed various strategies such as efficient capping processes, either by cap snatching, yo, shout out to the flu virus, cap snatching, or capping activities of their polymerases, shielding nascent genomes and antigenomes in the nucleocapsid, or efficient coupling of nation 5' and RNA with a protein that is necessary to prime the viral polymerase, picoviridae. In addition, they have developed many other strategies to counteract the IFN activation pathway. See this paper for review. Kind of a lackluster, not very exciting way to end the paper, but that is fine. And we are how far deep into the stream? We are what is the time code? 321. 321. This is for Astral's word count, and I did not include reading the methods section, just to the end of the discussion. Okay, that's more of a note for me so that I can make Astral's hobby a little more accessible, because I love that word per minute chart. It's so funny. Yeah, it does feel like they crammed a bunch of cool and interesting information from background papers all the way at the end for no reason, when I would have really appreciated the context, you know, at the beginning of the paper. So I'm exhausted, actually. This paper was difficult to read for reasons that the typical PLOS papers generally are not that difficult to read, right? I think generally PLOS papers are not as fun or easy to read as Nature or Cell. But I think this one is just an artifact of it being from 2007 and maybe a bit of language barrier, but I will say that, like, as I said when Myelin was around, the arrow of time points forward, and I think the standards for modern communication have risen. So the fact that I am so, like, tired of this paper is an indication that even though for the general public science remains very inaccessible, I think the fact that PLOS today contains papers that are a lot more readable is an indication of genuine improvement in the field, right? You can complain all you want about science being opaque. It is, but that's because you need to know a bunch of stuff before you can start learning. But the fact that I, as someone who is not a virologist, nowadays I can go into a PLOS paper and be pretty happy and pretty comfortable if it was published recently. This paper from 2007 was a lot harder, I think. Even though I don't think that anything in here operationally was that difficult, I think the communication skills of the average scientist has actually improved a lot in the past 2 decades. Should they rewrite papers from older times to meet new standards then? I don't know who you would pay to do that, or unless you ask ChatGPT to do it, but as we all know, it can be kind of problematic for it can be kind of problematic to ask ChatGPT to do anything these days, especially when it comes to making sure that papers are communicated well, accurately, and faithfully, right? Which is not necessarily a given when it comes to ChatGPT. Okay, so I am very, very tired, and I wish to stop talking. I hope you have all had a wonderful day. If you aren't already aware, I do have a Discord. You can type !Discord to join it. I have a new channel where you can see the schedule that will be updated month to month. I'm not like other streamers. I'm really not, in a sense where, like, I need to plan streams like way ahead of time so it fits into my video content creation schedule. So every beginning of the month I will publish a month worth of dates by which you can find me making content. The next stream I'm very, very excited for. It is going to happen on the 21st. That is next Saturday at 2 p.m. EST. This is a collaboration with the Octopus Lady, who we got together and figured out this really interesting question, which is, why doesn't tetrodotoxin poison the pufferfish? And what are the mechanism of tetrodotoxin resistance? And I think that's going to be really, really fun. It's a really, really interesting problem to solve. And the answer is, my understanding of the answer continues to evolve, because it's not super straightforward. And actually the people writing this stuff is, I don't want to give too much away, but it was very interesting how my assumptions about this topic have evolved over time. So I can't wait to share that with you, with the lovely Octopus Lady. You can go search her on YouTube. A much larger creator than me, so I'm like extra excited to get her on, just because I know numbers are not everything, right? But the fact that I have been acknowledged by somebody whose numbers are a lot larger than mine is a good feeling. So I am excited to see you there. So I also have another stream planned that the details got finalized. Is the day after I will be talking to a philosophy VTuber, Charis Euphranor. I never know how to say his name. Chairman, I call him. I will be having a philosophical discussion about the philosophy of education, which, if you liked hearing that in my stream when I was doing a week of streaming, I will have a pretty good discussion with him. We actually talked like 2 hours to plan this stream, and I think we have pretty good chemistry, and he's an interesting guy, and he's capable of asking really interesting questions. So if you're interested in kind of the hidden bones of my philosophy, I think that's worth showing up for. But with that all being said, I hope you all have a lovely day, and I will see you all hopefully next Saturday. Bye, y'all. Bye-bye. Bye, y'all. Bye-bye.